As far as I understand, the focus and scope of this Zoologica Scripta has changed over the years. While in the 1970s and the 1980s, large morphology-based taxonomic papers were published, but today the focus is more on systematic, phylogeny, evolutionary ecology, phylogeography, and conservation biology. As I am a microbial ecologist, I have to admit that this topic is very stimulating to me. And I have been working for 30 years already with looking into diversity of microorganisms, which is quite challenging, and have been very fascinating by the diversity of microbes and also the systematics. We started really with systematics on the functional traits, but then the molecular revolution came, and we started much more with genetic tools and the genetic approaches. So systematics, phylogeny, and also ecology is very, very important. I have to say that in microbiology, or studying microbiology doesn't make sense except in the light of ecology and evolution, and that's something we really learned for the last decades. At the COP26 meeting that was held in Glasgow in October 21, it was emphasized that we have to face two global crises at the moment. It's the climate crisis and the biodiversity crisis. And we can't ignore these two, or we can't handle these two separately. We need to join forces and think of both things at the same time. Also in April 2022, the 6th IPCC report was launched on impacts, adaptations, and vulnerability. And this report also recognizes the interdependence of climate, ecosystems, and biodiversity, and integrates knowledge more strongly across the natural and ecological areas. So therefore, I think the scope and the topic of this meeting today is very spot on and emphasizes what we need to have better knowledge on from now and also in the future. Again, as a microbial ecologist, I have to, since I have such a great audience here, I have to say that the microbial diversity is surely the outermost frontier of the exploration of diversity. The scale and mysteriousness of the microbial world have deceived some into thinking there is nothing interesting there. Even when Michael Rosenzweig wrote his paper in 1995 on species diversity in space and time, he said this: "I do not discuss the biodiversity of microorganisms at all. From the point of view of diversity, they are probably the most poorly known of taxa. Perhaps the diversity shows many patterns, but I'm unaware of them, and I don't want to discuss them in this chapter." Of course, this frustrated a lot of microbial ecologists, and as a result of this, we joined forces, and at least we managed to write a paper in Nature Review on microbial biogeography, putting microorganisms on the map, trying to link the macroecology with the microbial ecology. But we had to admit that Rosenzweig, he was very right in his statement. So the exploration of this frontier presents the investigator with very many multiple changes that we see, which are little by little being considered, restricted, and hindered. Although great progress have been made in the microbial ecology world, there seems to be doubt that the golden age of microbial diversity lies before us and not behind us, so we still have a lot to do. So microorganisms are not the oldest and most diverse living organism on the planet, but they also represent the vast majority of diversity, and that needs also to be taken into account when we study genes for evolution and ecology. And they've also been on the Earth for the longest time. The Earth has been 4.5 billion years old. 3.8 billion years been solely, the microbes were, the first microbes were present. 2 million years have only been microorganisms present on these. So that's a lot of evolution going on, going from a very reduced environment to an oxygenated environment. So this is something we need to emphasize on. 2 weeks ago, I attended the International Symposium for Microbial Ecology. Finally, we had a big conference physical people. It was fantastic to meet again and to discuss very small topics and meet with colleagues. And of course, a big debate where can we put the root of the universal tree? Can we put the root somewhere? And quite a few talks on this topic. I should take a very short version for you here today. Phylogenomic placement of eukaryotes is hard and it's leading to an extensive debate. So we're still debating where the root of the tree is. But one conclusion that I would like you to think of today is that we, the humans, the eukaryotes, the mammals, are a weird clade of the archaea. You should think of that during today's lectures. Okay, I think I put together a very fascinating program. I really look forward to be here and to learn what you have. And I wish you all the best. You're going to go into consequences for biodiversity loss, importance of good databases, microbe-host interactions, and taxonomy and systematics, which is really spot on what the knowledge we need to live on this world and to take things further. So with this, I open the symposium and I wish you luck with the program. Thank you.
Yes, is it working now? Yeah. Thank you very much for this warm welcome, and again a very big thank you that Norwegian Academy of Sciences and Letters is helping to organize that and housing us here today. So great opportunity to be together here today. So before we go into deeper discussions of biodiversity issues, Per Sundberg, where are you? Here?
Yes, I'm here.
Will present to all of you some further details on the Zoologica Scripta, who is also organizing today's symposium. And
thank you very much. Yes, you took my
welcome.
first words there. So this is a Scripta symposium, the third one, and we thought it should be appropriate to say a few words about the journal. So Zoologica Scripta is one of the leading journals in systematic biology. It has an impact factor just about 3. It's edited by myself, Per Sundberg, Lutz, and Per Eriksson, who sits over there. It's published on behalf of the Academy of Sciences here in Norway, and the Sciences and Letters here, and the Royal Academy of Sciences in Sweden. The first volume was published in 1971. But in fact, the story of this, or the history of this journal can be traced much further back. In fact, it can go back all the way to 739, when the Swedish Academy was formed, and one of the founders was Carl von Linné. And one of the first things that did there was to set up a series of publications, Kungliga Svenska Vetenskapsakademiens handlingar. I'm not sure how to translate that, but I think you understand what it was about. So it has a long history, and this turned into, it kept on going like Øvreås, it kept on going like Kungliga Vetenskapsakademiens handlingar. Then it turned into Archiv for Zoology, Archives for Zoology, which was also published by the Swedish Academy. The Archive for Zoology, it was a very traditional zoological journal. This is kind of typical paper, what it looks like. And for you in marine or invertebrate biology, this is the first report of this Xenoturbella, which is quite an interesting organism, by Lindblad in 1949. So this is very typical kind of what it looked like in those days, Archiv for Zoology. Then in 1971, it was decided that we should change from Archiv for Zoology to Zoologica Scripta. And from the Troy preface, and it says that it should be evolutionary zoology, covering aspects of systematic, structure and function, taxonomy, phylogeny, and biogeography. Evolutionary problems based on research within paleontology, ethology, physiology, etc. So Scripta will be more specialized than the periodical Archiv for Zoology, which will no longer appear. And this is the first volume, what it looked like in those days. It was published by a Swedish publishing company. In 1977, it merged with the Norwegian Journal of Zoology. I'm not exactly sure the reason for this, but I think it was considered that Norwegian journal was a bit too local in a way. So it was merged, and now also Norwegian Academy entered the owner of the journal. And in the 80s, it really became more and more a taxonomic journal. It went away from these evolutionary aspects, from morphology. It was very much, not morphology, sorry, but it was very much taxonomy. And this is a very typical paper, what it looks like. Species descriptions, the full of species descriptions, basically that all the volumes was about new species. In the 90s, there were new editors. It was me and Fredrik Pleijel who entered the journal, or I've been there before, but now I became the chief editor, and Fredrik Pleijel was executive editor. And we changed the publication profile and scope of the journal, and we turned into systematics rather than taxonomy, sorry, emphasizing phylogeny, and we also later added a subtitle, an International Journal of Systematic Zoology, to mark these changes. What we could see was that the impact factor, I think this is scripted. Straight away it went up, so we could cover much broader. We attracted more readers, and there were more cited papers, because all the taxonomic papers and new species are extremely important. My background is taxonomy, so I really appreciate the importance of new species. However, it doesn't really make citations. Which one can discuss? One could say the authors of names should be authors also as in reference lists, but they aren't. So, and also to mark this, we changed the cover, and this is a tree from Haeckel. Maybe the German people here recognize it. Haeckel was really advocating Darwin. So this tree is not based on what we would say today a real phylogeny estimate, but he wanted to emphasize that we should do the classification in accordance to the phylogeny rather than just what we think is right, wrong. So this is he produced a lot of trees for various groups. I think those in zoology here have probably read his papers, old papers, and really nice illustrations, but they were not based on any kind of proper estimation or based. And then later on we changed another tree here, which is from Charles Darwin's notebooks. It's the first written evidence that someone has been thinking about terms of. So that predated that tree, in fact. So this is from his notebook. He said, I think this is how we can describe the evolution. So this is the first written, published, or not published, but written evidence that Darwin started to think in these terms of phylogenetic trees. So we thought that was quite a nice illustration here to have on the. And I noticed I had a student. He had this tree tattooed on his back head. I like it. It's really nice. And now there have been changes in. The publishers, now it's Wiley. It has been started from the Swedish company Almqvist & Wiksell, then it was Pergamon Press, and for some time it was Springer, and then it was Elsevier, not Springer, Elsevier, it was Elsevier, that's right. And then it was Wiley, Blackwell, yes, and then Blackwell merged with Wiley, and then Blackwell disappeared. So. Okay, and what happened when we did this change of publication profile? We changed also the taxonomic coverage. So if you look at the 1991 here, there were 3% vertebrate papers, and invertebrates were 81%, 82%. And now it's down to invertebrates are now 37, and vertebrates is 37 around that, and 25% like spiders, and very few protozoans. But we also have sometimes more theoretical, technical papers. Just a few words for you young people here. The major shifts in taxonomy here which has happened is what changes when before phylogeny became sort of, for you it's obvious, it's self-evident that you're going to base your classifications on phylogeny. That was not the case when I started as a taxonomist. And the older people will remember the heated debate about the cladists and the non-cladists in the 80s. We were talking about that yesterday. We were in physical fightings at a symposium where people were really fighting about these. Unbelievable. But it was really a very heated debate about this. And in our case, the first paper, when there was the word phylogeny was in, the word cladistics. I don't think anybody used the word cladistic any longer. I don't think, no, we don't know. That was in 1990. And then, of course, the molecular data came into from when the major revolution there was when Kari Müller invented this PCR technique that really changed the field. And suddenly you can have even non-molecular biologists could do sequencing, could have sequences and base their phylogenies, base their classifications on those. And the first paper in Scripta with molecular data was in 1998. And of course, today, molecular data is standard. It's much more genomics today than it used to be. Another change we've seen over the years is where articles is submitted from which countries. It used to be real. I think this is Europe here. That's in this area period here, and this is really where the bulk of paper came from. And certain countries like Sweden, UK, we had a lot of systematics and taxonomy was big in Sweden going back 15, 20 years ago, and we had a lot of submissions from Sweden. Less so from Norway, but UK, Germany, France, Italy. And what we can see now is China. It doesn't really give a complete correct picture because now this is the first author, but still the trend is quite clear. It's China, it's Brazil, United States, and you see you don't even see Sweden here, United Kingdom here. So low submissions from those countries. And I can just see that in Sweden the number of systematists are declining in general. The area is changing. Right now, we used to, in Gothenburg, we used to be a group of 45 people, systematists. Now is really, we have no taxonomists right now, I don't think we have. So this is a major change, and this could also be a challenge in a way with changing publications, traditions, and cultures and stuff like that. But in general, it's China and South America, that's in the United States. Another thing which has changed is that from being a very solitary work to teamwork, if you look at average in the number of authors per article in 1971 was basically one per article. So it was a single author papers. Now it's last year it was 5.7 per article. And the reason behind this is that there are several reasons, but one is sample regulations. It's very difficult now to actually get permissions to collect animals in different countries and to export them, which means that you have to collaborate. I know that in birds, for example, Paris, that's a lot of collaboration. Basically, it's impossible to go there and collect anything. You need so many permits. So that's one reason. But the other reason is, of course, that today taxonomy is not just describing species or systematics. We need molecular biologists, we need bioinformaticians, we need geneticists and stuff like that. So it really is a teamwork in a completely different way than it used to be, which also can make it difficult to find referees to cover all these aspects. So in a single paper, there could be many, many areas of expertise really needed to cover that. And another thing which has changed is that now systematics and phylogeny, which Lise also mentioned, is really an integrated part of ecology, of other and phylogeny, phylogeography, conservation, stuff like that. This is really the core. And you see when a lot of discussions about what's the relationships, and so it's really a very basic thing which has to in many areas in biology today. So what will happen in coming years here for Scripta? What we can see now is that there's an increasing number of journals publishing, more competition. You probably read about these predatory journals. These are just, I could find hundreds of journals with biology in the title, for example. And some are good quality, others are not good quality. You have probably read about these situations where people sent in fake manuscripts and they got published within days, and there's just rubbish in the text. They are not peer reviewed. They are not at least checked. It's just a matter of money for some of these journals. There are, of course, very many serious journals in our area as well. There's also now a requirement demand on open access from granting bodies, which is also to make changes for us, how to deal with that, and how to pay for that. Now there are many agreements between countries. I'm quite sure that Norway has agreements with some publishers so that they cover your publication costs, but it's in Sweden at least, I'm quite sure the same in Norway. If you have money from a research council, you have to publish open access. There is no alternative, basically. But we have also in Sweden agreements with the major publishing houses, so it's covered, basically. There's also more new ways of publishing. There's more interactive ways of publishing. We have not gone into that field, but there are journals where you put in your paper, and then you can have comments on the paper, and everybody can comment, and then before it's actually final manuscripts there. So that could be. Let's also ask questions: who's actually the author of such a paper? So that's something which is happening. There's also pre-review. Journals. This was very clear during what you could do there. You publish your manuscript there. It's published and it's open for people to read, but it's not peer-reviewed. And it was very obvious during the pandemic. There was a lot of many studies published this way, got a lot of public attention, and then it turned out they was just crap, and they were never published in real papers. But for the general public cannot distinguish between this. I noticed in some of the newspapers in Sweden, they talked about this when these scientific journalists, they were aware of this, saying they should be aware this may not be a solid paper. But for many other people, this was good published papers. Another thing which is changing for us is that the research field, classical systematics, is in decline. It's something we have to deal with. And taxonomists, I mentioned before, is also in decline. We have very few taxonomists today, and I know that some paper talks later here will really emphasize the need for taxonomy in certain things. But this is some of these what it will happen in coming years that we will have to deal with. And tomorrow we have a meeting with our advisory council to discuss. The editors will have a meeting to talk about. We have I think you mentioned this, Lise, that the scope has changed over the years, but maybe we should change it once again. Maybe we should take. But what happens now is if you can see there, it's quite general focus, but scope. But there should be placed in the phylogenetic systematic context. So it means that I, as a chief editor, basically I reject 20-30% of manuscript because they don't fulfill this, even though they're really good papers. So this is something we have to talk about. Shall we change this to make it more broad? Must this be a requirement? Because now it's much more about not what is this, but more why is this? So we have much more that kind of very interesting papers, very important papers, but they don't have this particular systematic context. And it may be that we should think about that to take away that from the scope. We will discuss that tomorrow. We will also talk about the title of the journal. What does it? Zoologica Scripta. It doesn't really tell you what it's about. Well, it's about zoology, obviously, but what is about evolution, biodiversity, or whatever. So this is something we should at least discuss how to deal with that. Because if you're looking in the enormous amounts of journals where to publish my paper, you may not find the old people here, the older been around. They know the Scripta, but young people coming in here. I've done a paper on evolutionary biology, phylogeography, where to publish. They will not look for this journal. They will look at journals of phylogeography, journals of evolutionary biology, or something like that. So this is something we are thinking about. There are problems with changing titles, so it's not obvious, it's not that easy. But so that will be discussed tomorrow. So that will end my talk about this. And have you comments or questions? I think there's time for a few questions. If not,
thank you, Per,
okay,
for presenting the Zoologica Scripta and also the challenges and discussions that are going around that. I hope you all consider Zoologica Scripta an attractive journal. And so any other questions, comments, discussions or whatever, always possible during today's breaks, whenever. Either with our chief editor, Per Sundberg, or over there,
Per, you maybe can stand up.
And myself.
You can stand up as you...
So just contact us, and we are happy to answer questions, take up discussions, and so far and so forth. Okay, after this introduction of the Zoologica Scripta, maybe we go into the field of biodiversity. And it's very much a pleasure to welcome Cecilia Lindblad from Sweden. And she will review, I guess, for us quite a lot of issues that we have with the loss of biodiversity, recommendations, what we can do with it or what we cannot do with it, and certainly a good starting point for understanding what is actually the challenges with respect to biodiversity in the future. Thank you very much, and
thank you very much. And it's really a pleasure to be invited to this symposium on systematics and taxonomy. And I'm not at all an expert in this field. So my background is totally different. I have been in Stockholm University from the Systems Ecology Institute, and my PhD was about coastal habitats, so I have nothing to do with this. But however, the last years I have been working at Swedish Environmental Protection Agency with a question dealing with biodiversity on different levels of scales. And I also followed IPBES. So I can really see the need for more research in taxonomy, systematics, and investments in databases and big data for better understanding to reduce biodiversity and the loss of biodiversity and mitigating climate changes. And I will show you a bit about that. But first, a kind of overview of what IPBES is about. Yes, everyone here knows perhaps what IPBES stands for. And it's like you're perhaps better familiar with IPCC, and it's the same mechanism, but for biodiversity instead. And IPBES' mission is to strengthen the foundation for better policy through science, and for conservation and sustainable use of biodiversity, and human well-being and sustainable development. So it's a large part and quite complicated. And IPBES has existed 10 years, by IPCC is about 30 years now, I think. So that's why it's also more well-known. It's an independent intergovernmental body. And now it's 139 governments as members. And the work relies on financial and in-kind contributions from governments and experts. And that's also always a problem for this type of organizations. And you see here how it's organized, the plenary meeting, where everything is decided, where all the members are meeting nearly each year, and we have a meeting now this summer. And Norway is quite active here. In Trondheim, there is a technical support unit dealing with the capacity building, so it's a big connection with Norway. And all kind of information about the IPBES is easily to find on the web page. So bridging science and policy, I think that's really important and also really difficult. And IPBES findings should be policy relevant, but not policy perspective. And that's also really important, because the researcher here dealing with all these assessments, they give information that is placed on the hand of the policymakers. The most important thing that IPBES has is the conceptual framework that was founded also in the beginning. And you can see that there are this is the right way. You have different colors here. If you can see that, it's black, it's green. And this shows that IPBES is really in favor of connecting different type of science. The modern science that we talked about here, perhaps, what we do in Europe or in other countries. And then we have knowledge systems that is more local from indigenous peoples that also have an important role here in this information for IPBES. And this is, I'm not familiar with this apparatus. And all these boxes are illustrating in some kind different chapters in all the assessments. So it's the same group of chapters. We have the direct drivers, we have indirect drivers, we have the contribution to people, ecosystem, goods and services, nature gifts. This is the way of express how people interact with nature. And we in Europe and here, I think in Sweden and as well as Norway, we use ecosystem services but in IPBES they try to more and more use the nature contribution to people now. It's more inclusive and take into account the indigenous knowledge as well. And this is a base of the IPBES, so it's really important to see that. And the scope is dealing with the national to global level. Local level is always kind of small examples in the assessment. And the IPBES assessment process is it's a long snake like this with a lot of parts. And the first, it starts with the request and the scope. The request could come from the members of IPBES and also CBD and other organizations. And then we have the evaluation, and we have a lot of experts coming in to working with the assessment. Perhaps someone here in the room has been asked or involved already. And you know that there are a large group of people, could be hundreds or several hundred experts coming together and talk with each other, sometimes from different disciplines, which could be a hard exercise in the beginning when they start to talk to each other. They don't even know how to explain to each other the different fields they are experts in. And at the end then we have the process of reading the document and take it up on the plenary and then to accept it and continue with the scope of the processes, ending with supporting the assessment and try to convince policymakers to read at least a part of it. All the assessment has tried to show the confidence in the findings, so the readers know how sure they could be on different findings in the texts. And all the literature that are assessed is could be scientific literature, grey literature, and whatever of reports they could find in this. And also they have a level of agreement if the data in the literature are agreed or if it's a big difference and discussions between researchers in certain topics. So this is illustrating in the text. After all findings, you know exactly where in this box you find if it's inconclusive or it's well established, for example. So I think that helps decision maker to be sure if they can rely on what they read. So far, IPBES assessment has started. The first that come out was this one about pollinators, pollination and food production. And it was 2016, and this also was scenarios and models for that helps decision makers to show how they can go further with different knowledge. This is the summary for policymakers. So this is a document about 40 pages. Background document could be up to 1,000 pages. And now it has also been developed some shorter summary for a policymaker could be read when he's going in an elevator for 3 levels or something like that. Just 1 page. That's perhaps the most they have time to, but it's so important to get this out to people and to get use of the information, this compilation. And it has been, of course, a lot of assessment before about biodiversity, but this IPBES try now to include biodiversity and humans and well-being in the same document, and I think that's also really important. And 2018, it was the regional assessments come out for the 4 regions, UN regions, and also the land degradation and restoration. And 2019, we have this global assessment that was highly observed in media as well. Then we have some workshop report. One was on, of course, on pandemics and biodiversity, just to try to get scientists together and to discuss what effect and how the possible future for coming pandemics have coupled to biodiversity. And then we have the IPCC IPBES workshop on biodiversity and climate change. I will come back to that a little bit later. This year we have accepted 2 new assessments. One is dealing with the values of nature, to validate ecosystems and how to do that, the validation methods. We have used a long time, sometimes in Sweden, the economic validation of ecosystems and nature, but there are other methods as well. And it's a lot of methods, but yeah, it's interesting to read about how these are used and what are the outcomes from them. So you can, and the other assessment that was accepted this summer was a report about sustainable use of wild species. So welcome to go to website of IPBES and download those documents. And the upcoming assessment will be Nexus, more really integration among biodiversity, food, health and water in the context of climate change. So there you see the couplings between biodiversity and climate. It just started, so it will take about 3-4 years before it will be published, as well as the transformative change and business and biodiversity will be out in 3-4 years from now. So I will just show you a little bit of the results from the global assessment, where you probably followed the attention in media was quite massive, at least in Sweden. They saw it's terrible. 1 million animal and plants will be threatened and with extinction now. So that was really what was highlighted in media. But also that the wetland have been lost, and we have less biomass of wild mammals. And the most important that we have altered land and marine environment in such a high percentage. There's a lot of tables in the document, and I'm not going to show you all, just some few. And this is a global indicator of natural ecosystem structure. And what we can see here, that's the bigger change is seagrass meadow areas is globally has been declined since 1970. And wetlands and coastal habitats, for example. And also, this is a table showing the state of nature where we had the pristine area here at 100%. And all the blue spots here are marine habitats, ecosystems that are far from pristine now. So this is a type of data and compilation of data that is shown in the assessments. And what is doing this big change of our nature then? First of all is the direct drivers of change that have accelerated during the past 50 years. So 50 years is the time span that is almost the focus. And the direct drivers are then mainly land and sea use change, direct exploitation, climate change, of course, pollution, and invasive species. And they have separated in terrestrial, freshwater, and marine ecosystems. And you can see that there are a bit of all these direct drivers affecting those habitats. And if you look at the impacts on the global scale here for different classes, we had the genetic composition, for example, here. And here you can see how they explain degree of confidence. Inconclusive is marked here for genetic composition. And this is, you can understand then that more data and information and knowledge is needed here. So that's support more research in this topic, or more databases or accessible database that could be used for updating this. IPBES is planning to update or to do a next global assessment in some 5 to 6 years. So until then, perhaps we can fill up the degree of confidence to be at least established. Most knowledge we have of terrestrial areas and also species population is quite well established. What you also can see with all this color is that the direct drivers are about the same in all the groups here. So it's difficult to define on this scale exactly one driver. The causes of degradation in nature are the indirect drivers, of course. And the indirect drivers are demographic and social, cultural, economical and technology, institutions and governance, conflicts and epidemics. And that's how we live, what type of society we have, our overconsumption, how the governance are working, how much of the governance can change our way of in the social life. And we have, for example, conflicts. Here is also corruption, which is a big problem in other countries, more than here in the Nordic. So all these indirect drivers are, of course, important, giving our change in nature. So bending the curve. IPBES is also presenting in the global assessment different scenarios for the future. And business as usual is really gray and bad. We can't go on like we do. And with the increased effort of conservation, protection of nature, biodiversity, and if we can change our society, go for more sustainable production and consumption, it will be perhaps even better than we are in now. So we can choose which way we want to go. This is far from systematics and the topic of this conference, but really all your information and knowledge here can help to bend the curve through this transformative change. So we need to have a big change of the word. Knowledge gaps then. We need more data, inventories and monitoring. And we need we have gaps on biomes, Arctic, marine, for example. I think we have some presentations today here about Arctic areas. More information is needed. We have taxonomic gaps, big data on many gaps, and but genetic diversity is really important to increase data. We need to integrate the scenarios and modeling more to put data together. Of course, potential policy approaches and the indigenous knowledge and people and local communities. We need to have more methods to collect their knowledge. Then I have some short slides here about climate change and biodiversity loss. We heard in the first introduction here about the importance to connect climate change and biodiversity loss because they are interconnected, and so far they have been separate, and still they are discussed quite separately in Sweden by policymakers. And they are clearly interlinked. And there is a need to find synergies to avoid conflicts between measures targeting climate change and biodiversity. IPBES did, together with IPCC, a workshop together. And we in Sweden, together with SMHI, we produce a report to discuss this from a Swedish perspective. So this is in Swedish, but if you are interested, you can find it on our webpage. And some short results from our Swedish report is that we already see the change in seasonal distribution patterns that lead to new species interactions, earlier spring migrations, for example, and by migratory birds. And we have a longer growing season, shift in climate zone, and increased effect forest ecosystem, especially in the northern part of Sweden as well as here in Norway, I think. And we have problems with the specialists, the butterflies with limited dispersal potential. And in the aquatic system, we can see that already warmer waters affect body functions, food webs, and fish catches. So we have to find new ways to catch fish and new species. And also the brownification of water is obviously in Sweden giving effect that it changed the ecosystem because of the water gets so brown. So we need to discuss critical land use require perspective, system perspective. You have to see it in a whole system. And forest biomass, carbon storage, and or biodiversity is a discussion now in Sweden. It's really, the forest has come up as an important, we have an election soon in Sweden, and just this discussion about forest owners and how to protect or not protect the forest is big. We have also the agriculture for food. Of course, it's important, but we need also to conserve or to restore the biodiversity. coastal environment is for settlement or for erosion protection or for biodiversity. All this has to be discussed. And they are, of course, politically important to have nice solutions for short time, but we need to think for long time. To reach out to the general public in Sweden, I do a lot of work for that. We have an annual conference at Swedish Museum of Natural History yearly now, since 6 years. Now we have one in October in Swedish, so you are welcome. We have several Nordic people coming from Nordic countries to listen to that. We have done an anthology that tells about biological nature and the ecosystems in Swedish. And we also try to go to summing up in Swedish the IPBES assessment, so it gets easy for people in municipalities to take care, to really read the main conclusions from IPBES assessments. Yes, summing up. So the bridge is important, to bridge science and policy. And of course, support transdisciplinary research, even if it's difficult for different disciplines to meet in persons that not really talk the same language from the beginning. It's really important to come further in that type of research. And of course, biodiversity and climate change need to be seen together. And the system perspective is really important. And we also hope that IPBES assessments will be used more in policy decisions. And everyone needs to work together as possible as it is. So, yeah, I hope this is at least a positive end of a negative situation for the moment in the world. Thank you very much.
Thank you very much, Cecilia, for this presentation, raising and pointing out already quite a lot of important issues. Is there questions to Cecilia right away? Oh, there's at least 2 or 3. I just begin here because it's closest. Okay.
Thank you very much. Thank you for this very interesting overview. I have a bit of a critical question coming from, it's also a problem for me. I was an IPBES lead author for Belgium, so I'm very much in favor of IPBES. And I like your slides of the knowledge gaps, that we lack these basic taxonomic genetic data and so on. What I always find difficult is that our policymakers still found this basic data collection useless. And I remember a meeting we had on the Belgian Ministry in Foreign Affairs with one of the major focal points, actually, of one of our major conventions, I won't say which one, when we were saying, okay, we need to invest more in biodiversity monitoring if we want to support our partners in the South for to fill these knowledge gaps. He answered no, but come on, we're not going to start counting butterflies. So how do we convince even the policymakers that are supposed to support biodiversity monitoring of
Yeah.
of the fact that these reports will need data at some point?
Yeah, how do we support? I don't know. I try. And that's why I invite them to these seminars we have, and I got the same answer from our polity. Yeah, so it's so depressing.
But this communication in Swedish, doesn't that help?
Not that much. No, they say it's interesting, but then they go back home and do the ordinary business they always have done. Yeah, so no. No, it's not a fun answer, but that's a reality.
I guess that continues just the last question. You said for a better policy informed by science over the last 10 years. Do you really see a difference in policymakers, that they are more aware of the dimension of the problem? Do you see that there is a development, either positive or negative?
No, back and forth. I mean, I like example of the forest in Sweden is now really going back, because now is a critical time with they need the economical crisis and things like that. The forest owner want to have more money out of the forest, so now it's going back. So it's not the biodiversity and the crisis in nature is not overarching the need for money. So no.
So I suppose this is kind of all following on. I think there's kind of a tradition of skepticism towards policymakers. Yeah, so I was quite curious about, with the validation in the report, about this idea of kind of categorizing ecosystems by these like 4 different perspectives. And my impression was always that the reason that ecosystem services exist is just because it's a way for biologists to get a way for ecologists to get policymakers to listen to them, because they're often thinking in economic terms. And I was curious what your experience was with the policymaker response to these 3 other perspectives.
In Sweden, we use the ecosystem services concept. We haven't introduced really this nature contribution to people, really, and we hesitate like from our agency that we will introduce that, because then it's perhaps more difficult to convince them when we change. So this is more in IPBES family and in the group of research people that has developed that thinking. So yeah, but I think we should all be policymakers, perhaps. Then, yeah,
can we discuss that during the next break and just continue? Otherwise, we run already very early out of schedule. Another point I would also like to discuss with you, you mentioned these direct and indirect drivers.
Yeah.
I'm always wondering, isn't the ultimate direct driver the incredible increase in human population?
Yeah, that's really sensible to discuss and
it's
to have.
It's tough to discuss for various reasons, but is that it's tough enough?
Yeah. Yeah.
Reason enough, not really to have it on top of each slide.
No.
But this is our top challenge.
It has been debated many, many times.
Okay, so then we continue with some sort of a double feature. I don't know how you have arranged that with the 2 of you. Stine Markussen from the Norwegian Artsdatabanken and Hugo de Boer from the Natural History Museum in Oslo. They will introduce us to a Norwegian approach to mapping biodiversity, which is, I think your first
thank you.
thing that you will introduce the whole concept and the idea.
Of course, yeah.
We will see. Okay.
So me and Hugo de Boer are giving a joint talk, but we'll have split it into 2 parts. So I will start by telling you more about the Norwegian Taxonomy initiative, and then Hugo will come and give us the more broader picture of taxonomy and why it's important. I'm Stine Markussen, and I'm the coordinator of the Norwegian Taxonomy initiative, Artsprojektet, at the Norwegian Biodiversity Information Center. So, by starting off, I want to emphasize that knowledge about species is fundamental to understand biological diversity. And in order to understand nature, we need to be able to identify the different species, and for communication purposes, and to be able to share knowledge about species between us, we need taxonomic and systematic knowledge. And it's also a prerequisite for further and other research on species and ecosystems. And lastly, the knowledge of species is crucial for a knowledge-based nature management. For example, for conservation purposes, international commitments to protect habitats and species. So for 20 years ago, the Swedish government initiated the Swedish Taxonomy Initiative with the aim of identifying and describing all Swedish multicellular organisms. Inspired by the Swedish Taxonomy Initiative, 7 years later, the Norwegian government initiated the Norwegian Taxonomy Initiative in 2009, and the Norwegian Biodiversity Information Center were given the task to administrate and coordinate the program. And the Swedish and Norwegian Taxonomy Initiative has been collaborating since 2009, and there was signed a cooperation agreement from the start. In 2019, we also extended this cooperation agreement with Finland to enhance knowledge about species and ecosystems in the Fenno-Scandinavian nature. So, the Norwegian Taxonomy Initiative's main purpose is to strengthen the knowledge about species diversity in Norway to benefit research, management, and the public. And the long-term ambition is also for us to identify and describe all Norwegian multicellular species. And we have several sub-goals that I've listed here, that we need to have a special emphasis on mapping of poorly known species groups. And we want to build taxonomic competence and strengthen recruitment to the field of taxonomy and biosystematics. We will coordinate with similar projects in the Nordic countries and other species inventories in Norway. And we also promote DNA barcoding and disseminate knowledge about species diversity in Norway. And I will now elaborate how we work to reach these goals within the Taxonomy Initiative. Our main amount of funds goes into these annual calls for funding for mapping of poorly known species groups. And as you can see here, researchers and surveyors go out in the field, they collect material, and they go back into their labs and identify and describe and DNA sequence species. So from the start in 2009 and up until now, we have initiated a total of 120 mapping projects within the Norwegian Taxonomy Initiative. And the total amount of grants is almost 264 million Norwegian kroner. And from the graph here, you can see how or the number of projects for different universities, university museums in yellow, and research institutes in Norway, and also some from Sweden. And all of these projects have helped and been like vitamins to the scientific communities, where researchers can work systematically for a longer time period on specific species groups. But what do we get in return into the society and for management? First of all, the university museums get reference collections of higher standards, some comprising unique collections of certain taxa. We also get DNA barcodes, as the mapping projects also DNA sequence and fill up this national reference library of DNA barcodes. And this is important also for management. The mapping projects also deliver species information, pictures, and identification keys to our web page, Species Online, which is an openly accessible source of information to the public. And of course, the mapping projects are important to keep an updated register of species names. In Norway, we have Artsdatabanken, and it's important to have that updated. They also deliver occurrence data and species habitat relations that in total gives us a better knowledge about geographical distributions and habitat requirements of species in Norway. And lastly, the mapping projects also helps with competence building and recruitment to the field by that mapping projects include students, they include also postdocs, PhDs, technicians, and they are holding workshops. Some are also holding courses in collaboration with the research school in biosystematics. And all that increase the competence and recruitment to the field. Yeah, just briefly about the results from the mapping projects. From the start in 2009 and up until now, the mapping projects have in total identified more than 3,800 new species to Norway. And 30% of these are new to science, where around 300 has been described during their project period, and the rest of them are still undescribed potential new species. And you see from the graph here that there are a lot within the animal kingdom. And the main proportion here is insects, mainly Diptera and Hymenoptera. But there are also many marine invertebrates, such as annelids and hydrozoans, to mention some. And you also see that there are a lot of new species within fungi. Yeah, I also want to mention an assessment report that the Taxonomy Initiative funded by the Natural Museum in Oslo on the knowledge status of species diversity. And here 56 experts have assessed how many species do we know from Norway in different species groups, and also given an estimate on how many species we think we know we have, or we think we have in Norway. And summing this up, we know that we have almost 47,000 known species to Norway. But we also think we have 25,000 unknown species, meaning that we only know 65% of the Norwegian species and that 35% are still unknown. And this means that we still have a long way to go and that we need to keep having the Norwegian Taxonomy Initiative for a long time in the future. In the report, they also have an assessment on how much knowledge we have within different species groups on taxonomy, distribution, and ecology. But I don't have time to go into the details there. But there are still a lot of knowledge that we lack, and there are large gaps to fill within different species groups. At last, I want to mention two other things that the Norwegian Taxonomy Initiative are supporting. First being the Norwegian Barcode of Life, which is a network of Norwegian biodiversity institutions and individual scientists that are engaged in building a comprehensive library of DNA barcodes. And they are also a regional node in the International Barcode of Life project. And we have been cooperating with them since the start in 2009. where all the mapping projects have been able to DNA barcode species through this network. And last but not least, we also support and fund the research school in biosystematics, which are coordinated by the 4 university museums in Norway. And their ambition is to train a new generation of biosystematists to meet the society's needs for biodiversity expertise. And they also want to strengthen the research in biosystematics in the Nordic countries. And they offer a wide range of both practical and theoretical courses in biosystematics. And leaving that, I'm giving the word to Hugo de Boer.
Thanks a lot. Thanks, Stine. Right, I'm Hugo de Boer. I'm the director, the research director at the Natural History Museum. I've also been the head of the research school in biosystematics for Bio since 2015. I want to place a little bit of the work of the Artsprojektet in a broader perspective, and I'll do that with a set of slides that I will take you through. I think we should never be afraid to ask ourselves why systematics and taxonomy matters. This is a recurring question. When we talk to policymakers, they do not understand why it's important. Obviously, then we are failing to communicate why it is important. As we've seen, new species continue to be described, right? Not all organism groups we are describing as many new species as in others. Some groups, there's a lot to be studied. Some are easy to study. Some are very hard to study. The fact that we are not describing a lot of protists doesn't mean that there's not a lot of protists to discover. Whether we look at terrestrial ecosystems or marine ecosystems, we are describing new species everywhere. Also, the way the areas for which new species are described is not equal, right? We see that very few species are maybe described from Europe and North America, many more from South America and Asia. It doesn't mean that species diversity is much richer in those areas, but we know the species in Europe and North America quite well. There's a lot to be discovered there in Asia and South America. There's also some expertise there. And we have other areas such as Africa with only 12% of new species described within plants. And there we can assume that that is a lack of research. And why there's a lack of research, that has a lot of things are lying behind that. If we study species, and this is something that Stine was also into, we can also make predictions of where species continue to be discoverable, right? This graph, it's a study from Maura and Jets from last year in Nature, Ecology and Evolution. We see, for example, that the prediction is that there's relatively few species to be described among mammals and birds, whereas in the reptiles and amphibians, there's a bigger potential for discovery. We do not have this kind of data for all organism groups, but I'm sure that among the specialists here, there's people that know quite well in which groups there's a lot of diversity to be described. Species discovery, all right, it's not an easy thing. I think Per was in on that as well. If you have a journal and you publish one description of a species, it's not going to have a major impact. But some species discoveries are a great way of being able to communicate that hidden biodiversity to a wider audience. Also to an audience maybe that is increasingly disconnected from nature. If you discover a very tiny chameleon, then you should make sure that you use that opportunity and get out of it what you can. Also, I think earlier this year, just before the summer, there was a new giant water lily described from Kew. And this water lily had been in the collections at Kew for 177 years already. But the fact that you discover a new species, that it's the largest of all water lilies, then you have a responsibility as a taxonomist, as a systematist, to make sure that you grab that opportunity and that you communicate this wonderful diversity that we have. However, we can discover many things and describe them, but species loss far outpaces the rate at which we are describing new species. As Cecilia shared with us, the IPBES report establishes that 1 million animal and plant species are now threatened with extinction. That, again, is a clear message, but it's also worrisome because we are not, remember the graph, we were describing 15,000 new species a year. If we're losing 1 million in the next decade, then a lot of species are being lost without ever being discovered. Also, we know that wildlife is declining. These are just two graphs to illustrate that. We are losing mammal species at an increasingly alarming rate. At the same time, we're losing the habitats in which this species diversity occurs. For example, 32 million hectares of primary recovering forests were lost between 2010 and 2015. That's an area almost the size of Norway, and Norway is not a small country. Back to species loss and species discovery, these two maps here, the top map shows species discovery, so new species being described per country or per area. The bottom one shows species being rediscovered, right? You lose species, you find it back again. Where can we lose species? In areas where we have a lot of knowledge. Where can we rediscover species? Also in areas where we have a lot of knowledge. So we see that there's kind of hot spots on these maps that show areas where a lot of research is being done. That is great, but we should be worried about the white areas where we know very little. Species are probably being lost, but we are unaware of it. I think this was also mentioned by Cecilia. There are knowledge gaps, and one of these knowledge gaps is the taxonomic impediment. And the taxonomic impediment is something that you've probably heard within the context of the Global Taxonomy Initiative, which is one of the programs within the Convention on Biological Diversity. Taxonomic impediment, it's about lack of information about species, it's gaps in knowledge, and it's a shortage of trained people to describe that biodiversity, right? Often when we talk about taxonomic impediment, we use it a bit differently because we speak of a lack of taxonomic expertise in certain areas of the world, right? We have a kind of a north-south taxonomic impediment where we have a lot of material, a lot of collections, we have a lot of funding, and we have a lot of expertise in the north and very little in the south. The map that I'm adding here illustrates that a little bit as well. It's a map of the 200 most prolific fungal taxonomists that are responsible for the description of nearly half of the 360,000 species that are described. And you see that there's a lot of specialists. Sorry, I have to explain the colors. The orange are past people, so people who are no longer with us. The purple color illustrates people that are still there. So I think it was mentioned by Per, we are losing systematists, but we're also in a situation where systematists have been describing species in the north, but relatively little in the south. And a very positive development, what Per showed to us, is that the submissions coming into Zoologica Scripta, that we see a shift in the geographical range, distribution, right? We're seeing more authors from China, from Brazil, other countries, South America and Asia. That is maybe a good sign, because it shows that we are having a more equal distribution of taxonomic knowledge, even though we are losing taxonomists here. But Taxonomy. Taxonomy is, of course, to us, a clear puzzle piece in biodiversity. But what is the value of biodiversity? And that is something that we often need to express in economic terms to make people understand, especially policymakers. This World Bank report from last year made a careful analysis of the economic value of nature. And it concluded, again, a very easy conclusion to communicate, that protecting nature could avert global economic losses of 2.7 trillion per year from 2030. The figure that I've included here on the slide illustrates that this economic loss does not affect everyone equally, but people with lower incomes, they are disproportionately affected by nature loss. Also, as Cecilia was in on, the concept of nature's contributions to people, it's a good framework to communicate the value of nature to stakeholders. Whether it's too complicated, having 18 different NCPs, and whether it should be simpler, we don't know. But for some people, $2.7 trillion really goes home, and for others that want some more detail, they can look into this. Linking back to systematics and taxonomy, the Sustainable Development Goals, the UN Sustainable Development Goals, are a good way to show that. This illustration made by the Stockholm Resilience Center shows that these SDGs, they can be hierarchically organized, right? In order to have all the goals work together, the economic goals have to be met. Below that, the society goals have to be met. But at the basis of all of that are the SDGs linked to the biosphere. And especially SDG 14 and 15, life below water and life on land, that is where we, as biologists, as systematists, as taxonomists, make a major contribution. Because without that basic knowledge, we will never achieve the other Sustainable Development Goals. Too little, too late. Is species discovery a priority? If we have the species we need, yeah, how many more species do we need? Do we need to stick a label on every species? Do we really need to go and count butterflies? Yes, because maybe with burger and McNugget we have all that we need. What we know is that we're in an era with a very highly accelerated loss of biodiversity. We have all these worrisome curves that are curving up. We don't know what species we're losing. We don't really know the roles of these species in the ecosystems, but the ecologists among us understand very well the dependencies between species in an ecosystem. So to us, it's kind of easy to understand that this species loss could have dramatic effects. It's harder to communicate that to policymakers. This graph, you do not need to understand this graph, but the take-home message here is that only 2% of invertebrates, 7% of plants, and less than 1% of fungi and protists are red list assessed. What does that mean? It means that for the vast majority of organismal diversity, we have no idea whether they are happy, common, abundant, or whether they are threatened and going extinct. And we have to remember that policymakers, they use IUCN red list assessments to think about conservation priorities in areas. So it is a major responsibility to all of us that we work on red list assessment of species, because that helps policymakers do their work. whether or not it's going to erode the message of red list species assessment if not only pandas are red list assessed, but also butterflies that nobody cares about, that is another question, right? Because there's a risk in doing that. When we think of taxonomy today, we can see that taxonomy can contribute in new ways to science. Maybe we should all become policymakers, but I would argue that as taxonomists we should also stick to what we're good at, but try to be open-minded. In the Global Taxonomy Initiative Forum, which was held in 2020, a call of action was launched for the CBD post-2020 Global Biodiversity Framework, which will be discussed in the upcoming CBD COP in Montreal. One of the main messages there was that innovative and emerging technologies provide unprecedented opportunities for generating and sharing knowledge about the biosphere when combined with essential taxonomic knowledge, techniques, and skills. Yeah, so here we see taxonomists promoting the agenda of describing biodiversity and its needs, but also about being innovative about it. One of the ways to look at innovation is these three studies here that are sometimes referred to as turbo taxonomy. These are single studies that are describing a very large number of species, and they do that based on morphological data, but often in combination with molecular data. So by combining data, we can increase the rate at which we can describe biodiversity. Also, maybe to the surprise of some, environmental DNA studies. We can say that these studies have opened a Pandora's box of taxonomic needs, right? Because the more environmental DNA studies we do, the more we realize that we cannot identify the OTUs that we generate in these studies. We need taxonomists to describe the species, to generate barcodes that go into reference libraries so that we can actually use these methods appropriately. One example here is a study that we published earlier this year that shows that using environmental DNA from soil, we can assess plant diversity in the area. So instead of having a specialist going to assess biodiversity, which requires skills, requires time, we can go and monitor by taking soil samples. This can be done with unskilled labor, but it can also be done more frequently so that we get a better long-term monitoring of biodiversity. It's an example, right? But this can be done using environmental DNA studies in many ecosystems as long as we have good reference libraries. So, taxonomy tomorrow, what does the future hold? I think that we should be accelerating species description by complementing traditional morphology with molecular evidence, machine learning, deep learning especially. I think we can increase the rates of diversity monitoring to provide useful baselines to policymakers about the diversity that is there today and how it is changing in response to human-induced factors. And the balance here, I think also that we have to continue this trend of having a more equal sharing of taxonomic knowledge. And we need to achieve that through findable, accessible, interoperable, and reproducible data, through fair data. We can no longer sit on the knowledge that we have, but we have to share it with everybody in order to create kind of a democratic access to taxonomy. So then a question maybe with which I can end this talk to you: what skill set does a taxonomist of tomorrow need? Thank you for your attention.
Thank you very much to both of you, Stine and Hugo, and quite interesting question raised from you and into the discussion. Is there immediate questions? Or, yeah, Torsten.
We speak about the taxonomic impediment, and in the end, and also with Artsdatabanken, the goal is to build up competence and also recruiting into the field. However, in the end, it comes down to we still have losing of position at universities, of museums for taxonomists, and actually that's what a taxonomist needs. It needs to be a well-trained taxonomist first of all before it does all the other stuff, in my opinion. And so, is there any trend that this is turned around due to the activities Artsdatabanken does? So is there a way that we can say this dwindling of taxonomists at museums, especially at universities? It's not only in Norway, I know that in Germany the same. Is it turned around? So are we stopping that? Or are we still losing permanent positions of taxonomists?
I think that's a very good question, Torsten, and I think we've discussed that more often, but I'd be really interested to actually do a more careful analysis, maybe in collaboration with Artsdatabanken, to see whether these projects have generated permanent positions or maybe maintained permanent positions. I also think it's based on the data that we have with 4Bio, with the research school, we have kind of a good overview of which PhD students have been trained in the last 10 years. And we could look more at what kind of jobs that these people have gotten. Because I think, well, my first impression is that the Artsprojektet have created job opportunities, maybe mainly in temporary positions, as well as some permanent positions, but not often the pure associate professor positions. But that the funding and the competence that has been built up, as well as the DNA reference libraries, have led to a blossoming of consulting companies in the private sector that have been a big employer for taxonomists. You, Torsten. Yes.
Okay. Yeah, this is a very important point on training next generation of these taxonomic impediment. Can we continue the discussion during the upcoming break now? We are already a bit behind schedule, so it will be a bit shorter than scheduled. But enjoy the coffee, discuss what we heard, and see you back in 30 minutes.
All right, welcome back. We continue the symposium with two more speak before lunch, and the first one of them is by Veronika Johansson, who is a node manager at the Swedish Biodiversity data infrastructure and employed by the Swedish Museum of Natural History. Unfortunately, she couldn't be here physically, but she's on, as you can see there, she's sitting waiting. All right, so go ahead, Veronika.
Thank you, Per. I'd like to start by thanking for the invitation to have a talk here. And of course, I'm sad not to be able to participate in person, but now when both my sons have gotten COVID, it felt like a bad idea to go traveling. So at least we could solve it in this way. So as Per said, I'm Veronika Johansson. I'm the node manager of GBIF Sweden and also the project coordinator of SBDI, Swedish Biodiversity data infrastructure. I've been asked to have a presentation about the power and potential of biodiversity databases. It's perhaps a bit hard to separate these two, but I thought I would go through the more technical background when talking about the power of biodiversity databases, and then focus more on how and for what biodiversity databases are being used when talking about the potential. And finally, I will talk very shortly about the possible limitations of biodiversity databases before I summarize. I hope you can hear me okay. Let me know otherwise. What is a biodiversity database? It can really be quite broad and span many types of data. It could really be any kind of information that is derived from a living organism. From the organism itself, of course, with like observation specimens, but it could also be derived information such as genetic data, organism traits, or even vegetation patterns. And there's, of course, a lot of different biodiversity databases, and these are often thematically specialized, like, for example, some by region or by taxonomic group. But since I mainly work with GBIF, I thought I would focus on this network and use some examples from within this community. And GBIF aggregates data on organism level, mainly, you could say. So it's observation specimens and now also molecular data. I think most of you have heard about GBIF before, or the Global Biodiversity Information Facility, as it stands for. GBIF is by far the largest international aggregator of biodiversity data. And there are now more than 2.2 billion records available, freely available for anyone to use and download. It took about 10 years to reach the first billion of records, and that was reached, I think it was in the end of 2018. And now, just a couple of years later, we've surpassed 2 billion. GBIF only handles open data, and all data are published with a Creative Commons license, only open Creative Commons license. And you can access data through the web portal, but also through the R package, RGBIF, or through the APIs. To aggregate large quantities of data in biodiversity databases, it's of course necessary to use common standards for publishing data. And GBIF uses a standard that's called the Darwin Core Standard. And in fact, most biodiversity databases use this standard, or they are at least compatible with the standard. And in GBIF, you could say that there are 4 types of dataset classes that can be used to publish under. So it can be described as 4 different protocols of Darwin Core Standard, really. So depending on what type of data you want to publish, it can usually be fitted into one of these categories. So you have dataset metadata. It could be species descriptions or a method, for example. It could be species checklists, like the red list or invasive alien species lists. It could be more occurrence data, or it could also be more structured sampling event data. And the current data model in GBIF is currently based on the occurrence data, but work is now underway to expand this data model to cover more dynamic data. So you could say that there's an ongoing shift towards more sample event-based data. To illustrate the potential of the data available in relation to perhaps what could be available, I thought I would go back to the basics a bit and use museum specimens as an example. Museum specimens are one of the types of data available in GBIF, and it's really the backbone of GBIF and how it all started. But anyway, there's now around 206 million specimens registered in GBIF, so it's quite a small fraction if you look at the whole bulk of data, but still quite a lot. And what the information you usually can find is what species was found and where and when was it found. But there's also images sometimes, or other kinds of information available. And sometimes there are also this enriched information available, like coordinates that have been added or geo-referenced based on, for example, the label, if there's a locality name label. But the next slide here will show you what usually could be made available for a specimen, but not necessarily is made available. So this image is from DISCO, the Distributed System of Scientific Collections, and it illustrates the so-called simple digital specimen. And it shows how a physical specimen links with other artifacts and information that's derived from the physical specimen itself. So in this rounder, greener box, you can see the various Darwin core terms that are used and how it links out to other types of information. I don't have time to go into details, so what I again want to highlight with this image is the huge potential of museum data and the great addition to future research possibility it contains. I'm sure most of you have heard of these buzzwords before, at least the FAIR principles, which stands for findable, accessible, interoperable, and reusable. But there's also this these CARE principles, which is perhaps a less intuitive abbreviation, but it stands for collective benefit, authority to control, responsibility, and ethics. And these principles are more directed to give credit to indigenous and local knowledge. These principles are a great tool to use and to think of as an organization, as an individual researcher, or really anyone who collects or handles biodiversity data. And these principles are a large part of how GBIF also works, not just when it comes to how GBIF handles data, but also like how the organization works in general. So what I want to come to with that is that true potential both power and potential comes when data is freely available and easily accessible for all. It is also a clear benefit, of course, that data is aggregated at a central point for the users. It's crucial that data is standardized with common international standards, and of course that they are reusable and refindable. And perhaps most important, that there is a transparent process. I think transparency and openness creates these feedback loops through the data lifecycle. So you have to keep in mind that data is not a static resource. It changes over time and is constantly added to or improved or corrected. So the same logic applies really to like a published resource, like a book, for example. You have new editions where things get corrected, and the same applies with the data set. It's just that you have different versions of the data set. Another thing that I haven't mentioned so far is the importance of metadata. Of course, all data are really knowledge artifacts. So you might know everything there is to know about your data, but when you share your data, you have to make sure that the information you have about the data is also transferred to the potential user. In GBIF, all data sets and even the filtered downloads are provided with stable DOI, and that means that if cited correctly, anyone can go back to the exact data used in a specific research study. They could reuse the same data to reiterate the same analysis, or as new data is constantly being added, you could go back as well and use the same filtering options, but with additional data, and then do the same analysis again. So it's really reusable and refindable here. The diagram here shows the number of peer-reviewed papers using GBIF-mediated data, and as you can see, it's been increasing quite a lot lately. And the green columns here illustrate how many of these studies that use or cite by using DOIs. So it's quite impressive that it's now more than 3.5 papers published using GBIF-mediated data per day. To sum up this section, I thought I would read this quote by a co-author of this study here mentioned, which was featured in the GBIF news blog, because I think illustrates the power and potential of the biodiversity databases quite well. So here it goes. Without free and open access to massive amounts of data, such as those made available online through GBIF, no individual researcher is able to contact every country, every museum, every scientist holding data and put it all together. So this research would not be possible without GBIF and its global community of researchers and volunteers to make their data freely available. Over to the potential of biodiversity databases. And as I said before, it's a bit hard to separate the two, but here I thought I would focus more on the on how and for what data is being used. This is from a study made by Hebling et al. From last year, where they have investigated whether data integration enables novel research. They've used more than 4,000 studies as a basis for their analysis, and these are all studies that have used GBIF data, of course. And this map of science illustrates the various research fields in which biodiversity data is being used, and it's also illustrating the cross-disciplinary breadth of the usage of GBIF data. They've used some kind of reference base map of research fields. I don't exactly know, I don't remember what it was called, but there's apparently some kind of reference data map. Where more similar research fields are clustered closer together. And they plotted this with GBIF mediated studies. So here, the GBIF studies are represented by colored circles, and the size of each circle illustrate the proportion of studies available within that research topic. So as you can see, it really covers a large variety of research fields, from brain research to electrical engineering, really. But perhaps not surprisingly, biology is, of course, the largest field. This graph is from the same paper by Hebling et al. And here you have here they've used this correlation network analysis to visualize research topic clusters. Topics that are clustered nearer to each other and are connected with this gray line here are more likely to appear together in a given study. And this graph also shows what's hot and what's not. The redder a topic is, the more growth we see within that topic, and the more number of studies you have. The size of the circles here also illustrate the number of studies available within each topic. So, for example, you can see that there's a smaller number of studies on the species interaction topic. I don't know if you can see my arrow here, down here. But at the same time, it's also a topic that is on the coming and growing a lot. Other topics that are growing in number of studies are species distribution models, conservation and climate futures, and disease. It's perhaps a bit interesting that disease-related topics here cluster together here with the invasive species topics. GBIF centrally has responded to these results by initiating specialized help desks within some of these growing topics. There's, for example, a specialized support desk for health-related biodiversity data, and with the coming new project, I don't know if you've heard about this biodiversity digital twin. GBIF will also be a part of this, and there will be more support and use cases available for species distribution topics. I'm only going to go through one example next, but because I don't have that much time to go through a lot of examples, but if you're interested, I recommend you to have a look at the GBIF Science Review, where they make a summary of the research made available by GBIF. And to illustrate the growth of usage, I can perhaps mention that the previous science reviews, they've usually handled or covered 5 topics. So now you can see it's almost 20 topics available. So I thought I would present one really neat example of how biodiversity data can be used in an innovative and interdisciplinary way. And it's this tool that is called Biodem. It won the Ebbe Nielsen challenge last year. And this is a prize that is awarded to innovative applications of open access biodiversity data. It's an open source application that addresses the knowledge gap about the effect of society and political systems on the collection and the public availability of biological specimens. So very, very simply put, what they've done is to cross-reference two databases. You have the occurrence records from GBIF, and then there's this political systems database called VDEM. And this image that you see here is a screenshot of an interactive scatter plot where each bubble here represents a political country. The size of the bubble indicates the number of occurrence records available for that specific country. And the colors here represent the different political systems, where you have yellow color representing liberal democracies and purple are closed autocracies. And you can customize both of these axes. So if you want to try it out yourself, you can go to bio-dem.search.sh. I've written the link down here for further reading, I think. But this is just a default plot where you have freedom of association and freedom of expression on the y-axis. And just for reference, I've put in a few countries here. So I have Denmark, Hong Kong, and North Korea. This is another example. Oh, sorry. This is another example where I've chosen freedom of movement against the number of records available. And same reference here, so Denmark and North Korea as reference. You can see that there's, with higher freedom of movement, there's also higher diversity available. You can also have a look in more detail per country. Here is a screenshot of Cambodia to the left and Norway to the right. You have the number of records on the y-axis here, but please note that this is on a logarithmic scale. And there's a year here on the x-axis. And again, these are the default values in this application. And you can customize these diagrams as well. So go in and try it. It's a really neat tool. And some of the questions that they aimed to explore with the Biodem was, are democracy superior in record collection and mobilization? Which dimensions of political systems are important? How do armed conflicts impact record availability? And there's so many aspects to this application that I can't discuss now. I just don't have time for it. You should go in and have a look yourself, but it is quite apparent when comparing these two countries that it is quite clear that freedom of expression and political systems and also conflicts have an effect on record availability. Perhaps we can go back here again if you have questions later on. Before I summarize, I will just very shortly talk about the limitations and constraints of biodiversity databases. There's, of course, biases available in data, and it's sometimes hard to come around, even though it's something that GBIF works actively with. But when it comes to temporal biases, for example, it's hard to go back in time, collect more specimens. We have, of course, much more data available from present time than we do from, say, 100 years ago. This graph is from today et al. And it illustrates the taxonomic bias in GBIF. Here, the 0 value on the scale illustrates the threshold value, where the number of records available per taxonomic group corresponds to the number of species that should be available within that specific group. And as you can see, there's a huge overrepresentation of birds in particular, which is, of course, great for those who do research on birds. But there's also a huge underrepresentation of occurrences, especially when it comes to insects and various fungal groups. Another interesting aspect of this illustration is that it is mismatched to where the most research is being conducted. It is, for example, comparably more research being done in entomology and mycology than it is in ornithology. And this has, of course, changed with the evolution of eDNA, and I think these gaps will more and more be filled thanks to the incorporation of molecular data in GBIF. But it also highlights the importance of researchers to share their data much better than they currently do. This is a heat map of GBIF records. The more yellow on the map means that there are more records available, and it's quite clear that there's a bias towards the Western countries. There's, of course, several reasons for why we have these patterns, but this is at least something that can be worked on and improved. But there's, you can see the blacker areas where we have geographical gaps. When using big data from biodiversity databases, one have to keep in mind that the question you want to investigate determines the data you can use. So it's always context dependent. And it's really important to filter and clean your data. I've come across some data owners that are a bit hesitant to publish their data. They say that it's not perfect. They want to fix this and that before they share the data. But if you're in this position yourself, don't worry so much. You can just share the data. It doesn't have to be perfect. With big data, patterns usually become apparent anyway. They sort of shine through. It's also, there's a huge community available, so there's a lot of data owners or data users who are more than willing to help to clean your data and to give feedback with corrections and so on. But to end this section on a good note, I thought I would mention co-data statements of GBIF in their independent 20-year review. Stating that GBIF is the most comprehensive, openly available, application agnostic, easiest to use, and modern access points to known digital occurrence data. So to summarize, I would just like to again repeat the principles: be fair and care. I saw that there's another presentation later today that also discusses the topic. Openness and transparency, it's crucial for biodiversity databases and the success of these. The power and potential of biodiversity databases lies in the usage of the data, of course, not the volume of data. But it also resides within the community around biodiversity databases. So it's a great resource to tap into, and there's a lot of support and help available. And finally, I just want to urge you all to publish and share your data. You can help with the repatriation of data, you can add to future knowledge, help policymakers in having suitable background information, and of course, fill data gaps. Thank you, that was all for me.
Thank you, Veronika. It's amazing the speed of the growth rate of records in GBIF.
Yeah,
really amazing.
Mm-hmm.
And the potential, of course. We have room for a couple of questions for Veronika. Anyone? Yes, please.
Thank you. I'm noticing that iNaturalist and perhaps other citizen science apps, I don't know, the moment that an observation becomes research grade, it also appears on GBIF. And I'm personally a very avid fan of iNaturalist, but I hear a lot of professional scientists complaining that 2 observations or 2 confirmations is not enough for research grade and so on. What is the opinion in the GBIF community? Is that quality normally of these citizen science contributions appreciated, and is the importance of it growing with respect to the totality of GBIF?
Well, I think GBIF appreciates iNaturalist very much, and it's very few. I don't know of any other citizen science observation system that has this type of community that helps to validate records like iNaturalist has. And of course, to have 2 validations might be too few, some may say, but at the same time, data is not static, as I said. And the same goes with the records available in GBIF that comes from iNaturalist. So when people, I'm myself an avid user of iNaturalist, and I also help with identification. So the data is from iNaturalist. I know it's being updated every month in GBIF. So that means that even though you have 2 validations of the record, and someone might come in at a later stage and say, this is wrong, it's not this species, you're still able to go in there and suggest a correction. And with the next update of the iNaturalist data, this correction will be included. So it's an ongoing process of improvement as well. I don't know if that answers your question.
Yes, absolutely. Thank you.
Yeah, another one. Okay, if not, thank you again, Veronika, and stay healthy.
Thank you, thank you. Yeah, since I'm not going to be available for the rest of the symposium, I added my contact information here, so you're welcome to contact me with any questions later on.
Yeah. All right, thank you. Next speaker is Torsten Struck. He will speak about understanding biodiversity change, the potential of combining genomic data and taxonomic challenges like cryptic species. Torsten is a professor in evolutionary genomics and curator of helminths at the Natural History Museum here in Oslo. So, please.
Okay, first, thank you for inviting me to the organizers, and stay with me 1.5 hours. Then we have lunch. I hope you're not too hungry. And does it come now? Yeah, no. We had that before. We're in a world of crisis. We have climate change, we have biodiversity loss. I wanted my start my talk with some dramatic pictures. We have that heard enough. But as Veronika just mentioned, we are also in an age in biology where we have actually a revolution going on. We're in a big data age. Physics has been through this for some time, and now it's also hitting biology. We have seen that with the databases. Morphology also is going to big data changes. There's a lot of morphological data generated really quickly, and it's definitely true for sequencing. Sequencing costs is shown here. They have dropped dramatically they actually go beyond the Moore's law, which was about computational costs. And it's still dropping, it's slowed down, but it's still dropping. You have to have in mind the still logarithmic scale. And this is mainly due now to the newest development, which is single molecule real-time sequencing. They generate long reads, highly accurate ones, read one by now already. And this was, especially this one, it's a PacBio Hi-Fi, it was a game changer, and we will see that with some data we generated. These data, and especially the mini-iron, which you see here, that's the size of the sequencer, have now also changed what we can do in the field. This is a protocol which came out last year by Aaron Pomerantz and co-authors, and it's about using a metabarcoding approach. They do DNA isolation, amplicon sequencing of a barcode, nanopore sequencing. As I said, this is a sequencer, and then downstream data analysis on a laptop. It's all within the field, using miniaturized scientific equipment, and then without reliance on internet connectivity. So you can really do it almost everywhere. In the deep sea, it might be a little bit problematic, but in other cases, it might be relatively easy. And it takes 10 hours, so a working day, roughly, in the field. So there's huge progress in there, and it works. There has been another study which used it. They didn't use a setup of Pomerantz because that was published before. By Srivatsan, they collected insects in one Malay trap for 8 weeks in Uganda. They found more than 650 forage species. You have others to ask you, maybe Beate, what a forage species is. I don't know, but that's no way. They state that exceeds the numbers of known forage species currently described from the entire Afrotropical region. And this is just from a single point in Uganda. They had a higher accuracy in their barcodes with very little ends in the sequencing. This shows you now how accurate mini-iron has become as a system. They did morphological examinations to match up what they found with the molecular data, which is called here. That's called a molecular operational taxonomic unit. If it matched up with morphology, and it did quite good, 93%, and above 99% of the specimens they looked at. And most of this belongs to a huge, mega diverse genus, and which has been often neglected. So that works. However, barcoding, and maybe we will hear that a little bit later, has some challenges and some limitations, especially when we want to go maybe towards more ecology with abundance measurements. And maybe the next steps is doing metagenomics, as they do in the prokaryotic world already quite extensively. They more or less went to sequence genomes and nothing else. And that might be the next step. But if we want to do that, we are still then faced with the same change we already have now with a reference database, and we need to build that up. And there has been now proposed an initiative, it's called Earth Biogenome Project, by Harris Lewin and authors, and in 2018, I think, it was published in PNAS as a perspective paper. And the goal is, it's called the moonshot of biology, reference genome at chromosome level for each eukaryotic species, or almost chromosome level, in the next 10 years. That sounds very ambitious, and maybe it's not possible, but it's on its way. So EBP is established, and it's running. And there are now initiatives going on. And I don't know how aware you are of these. The first one to come out at the national level was Darwin Tree of Life by Sanger Institute. They have the goal to sequence 70,000 plants, fungi, and animals and protists in the British Islands and Ireland. So they are really hitting the road now, and they are setting standards. But Norway is also really early on in the game. It's the Earth Biogenome Project Norway that has been now funded. And, oh, sorry, now we go back. There. It has been now funded. It started last year, but it really hit the road now this year. There's 11 institutions all over Norway distributed, also commercial institutions or companies. The goal is to sequence Norwegian and Arctic species with an emphasis on marine species, but also to coordinate with the other Nordic countries and also other international projects going on, like ERGA. And several genomes are already underway. The plan is in the phase 1 to sequence 150 species from Norwegian species. And if you want to know more, you can speak with Kjetil. He is the leader of the whole project. He initiated everything after Levin's talk here in Oslo. And so it's his idea, and he's pushing that forward now. He's not here today. If you want to speak with someone else, Ule is sitting back there. He's also involved in the project. Then we have the also that all on the European level. This was started out of the Vertebrate Genome Project, and EVP also started out of the Vertebrate Genome Project. And here it's also that there should be the same on the European level. So a reference genome atlas for Europe. And there's the same goal as for all these approaches, but this was bottom up. It was built up from the bottom. It was the scientists coming together, and it gained a lot of momentum in the last 2 years. Now we are over 600 researchers from 48 countries, and it includes all 27 EU countries, as well as in Norway. This is how it looks. It's more a society than a consortium. We have a general assembly. There's a council where there's country representatives on. The most important part of it is here the scientific communities, where we start to harmonize procedures across Europe, standards, but also talk about which species should be sequenced and to avoid redundancies. We have an executive board, which is chaired by Camilla Mazzoni from Potsdam. And then we are building up an advisory board, but there's also then connections out. Sorry, first one thing. We started a pilot study, which was based on like bits and pieces here, different projects here, some sequencing companies pitched in, and there are now already 100 genomes for different eukaryotic species on the way. Some are finished, some are still going. We also coordinate us with regional initiatives like DTOL or EBP NUR, and we are also reaching out to the barcoding community. So, for example, the Bioscan initiative. And I will explain what this BGE means in a bit. As I said, it also has a national representation, and there is what we call ERGA Norway, so it's a national group which is part of the General Assembly and has 2 representatives as a council. In the moment, we are 37 members from 15 institutions. One point I want to mention here is, there it comes. We are an open community. It's not closed, it's open to everybody, and we welcome everybody. So please join, both at the national level as at the European level. We would like everybody who's interested to work with genomes at one way or the other. Taxonomists, bioinformaticians, conservation biologists, whoever could think we would be really appreciate if you come, because our strength comes from that we work together at all levels. This is the committee work, and you see here we have the whole process pipeline, which is related to genome assembly, genome annotation, so the hard work, but also here sampling. Standard operating protocols of how to sample best shall be, for example, established. And we also look at ethical aspects and different aspects, training, especially in the ethical aspect. When we speak about doing a project across Europe, Nagoya comes into play. And that's not an easy task, even in Europe, which you might think it should be easy. And again, it's open. Everybody, if you're a member of ERGA, feel free to work in the committees. You are very welcomed, actually. We have not that many who really actively drive things, so the more we have, the merrier. If you have questions, ask Rebecca. She's also here in the auditorium, and me, because we are the Norwegian representatives on the council. Now I come to BGE. So it's Biodiversity Genomics Europe. The aim is to accelerate the use of genomic science to enhance understanding of biodiversity, monitor biodiversity change, and guide interventions to address its decline. That's what we set up. It was in response to a Horizon Cluster 6 call, which actually asked for the implementation of EBP node, as well as for Bioscan node. And Bioscan Europe and ERGA decided we go together and join forces, our joint venture, and now we are together in one big project, which is called Biodiversity Genomics. So it's a barcoding community and the genomics community working together, not against each other, but in collaboration. It's a huge consortium. It's 33 institutions from 21 countries, and from Europe, from Norway, there's UIO involved as well as NTNU. NTNU is on the Bioscan side. UIO is on here, on the Earth Biogenome Project. It will start next month. It has a runtime of 4.5 years, and the aim is about 400 to 450 species to be sequenced. It depends a little bit on genome sizes. in different tasks. We have case studies, we have sequencing of biodiversity hotspots, and we have what we call community sequencing, where the community makes suggestions. So there might be an option for you to get a reference genome of your species of interest. It's not guaranteed. You have to go through a selection process. Again, if you want information, ask Rebecca or me. We are the ones who are involved here in a leading position from the Norwegian side in this. Then I want to make a little bit of advertisement now for a conference which is coming up, the first ever Norwegian Biodiversity and Genomics Conference. It's next year in February. It's organized by Øvreås Norway, EBP NUR, and the Cost Action G-Bike, which aims to bring genomics towards conservation biologists and practitioners. Therefore, we have also a pre-conference workshop on conservation practitioners. So if you're interested in biodiversity or genomics, save the date. It's in February, beginning of February. Again, if you have questions, ask Rebecca Kjetil from our side, or me, or Alexander, or Just about the conference. Okay, so why do we need the genomes, except for being a reference genome for meta-genomics? There's a lot what genomes can also tell us for monitoring. Here's a local study from Oslo, from Kjetil's group, on the Atlantic cod. When they looked at the genomes of these ones, we see there's a clear population difference between the northwest and the northeast Atlantic. That's maybe not so surprising. But then they looked into each of them and found there is also a clear difference between migratory and non-migratory groups or populations. So they were clearly separated from each other. They went a little bit further and looked on where they are different in the genomes, these populations. And the interesting thing, it's not distributed all over, it's clearly linked to certain what's called linkage groups. It's almost chromosomes, to certain chromosomes. In total, it's 4. You can see that here. The interesting thing is, in all of these, it's always in major inversions where these differences are. So these major inversions of the genome fix the differences you see between the different groups, between migratory and non-migratory behavior. It includes hundreds of genes and facilitates the co-evolution of these complex character traits, but it also allows to facilitate efficient and reliable ecotype identification in the field, because you can aim now for these differences there. You also can monitor more effectively on harvested core populations, if they come from a migratory or a non-migratory population. A similar example is here on the Iberian lynx. This is a huge author list, as you can see, for the genome. They showed a series of severe population bottlenecks, which went down since 50,000 years ago. But they also showed several signs of genomic erosion, reduced rates of weak to strong substitutions, low genome-wide genetic diversity with long stretches of regions of homozygosity. So there's these genetic erosion signatures and a high frequency of potentially deleterious variants. It's not a good sign for a threatened species if you have genome erosion. But in follow-up studies, other authors use these then to develop new markers to be able to monitor this more efficiently. They used snippet database. This is different sets of new markers, and they were better at individual identification, parentage assignment, relatedness estimation, ancestry admixture estimations, all of this which are necessary for an effective monitoring. So they have a more powerful, efficient, and flexible tool for genetic management of the threatened lynx populations. And as with the cod, you see, so the genomic data could be used to develop more tailored responses, monitoring tools to see at these endangered species. Okay, these are now all vertebrates. It's easy, they're nice, big, they have a lot of material. What's with now smaller animals, not so easy animals? This is what we are doing right now in my group. This is data now from the PhD thesis of Alberto Valero Garcia. We worked on a nematine. This is still in 10 to 20 centimeter size, so it's quite large, but it's very slimy. So that makes it really hard to work with them. But with BacBio Hi-Fi, we could actually manage. We got here a nice profile with high coverage. Smudge blot showed us it's a diploid organism as we expected it. Our blob tool showed we have here really nicely big contacts, which match to all different kinds, not necessarily nematines. This shows you again, we need to build up to have a certain identification because there's not many nematines genomes published. Actually, it's only one. And so the most matchup is something like annelids, mollusks, echinoderms, chordates, what's there for genome. If you look here now at this snail blot, you see we have really high numbers, and essentially the ones who know genomes, so the longest scaffold here is 13 million base pairs. We have an N50 value of 10 million base pairs, which is really good, given that the genome size is 160 M. The N95 is even better, in my opinion, 7.5. Also, when we look at the scaffold, we have only 95 contigs, and we did not do any high C on this. It's just hi-fi data. We have an L50 of 8 and L75 of 12. So even so, it is not absolute chromosome level, it's almost chromosome level data we got there already, just with a high coverage of 5. And we got also a good completeness of 96% of Busco score. So this is also probable now for almost any organism. But there are challenges, and we come to this now. We have also, the majority of animals is small. It's less than a centimeter. And we have here one, which is a gastrotrick, sorry, there. No. 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 It's here, this one, Lepidodamella squamata, it's 200 micrometers, so a fifth of a millimeter long. And we worked on a single individual. And we got a very good genome out of it. It's not as good as the one I showed before. It's in collaboration with colleagues from the US, Nick Roberts and Kevin Cosart. Again, we find here relatively large contigs, and they match against chordates and arthropods because there's even less available for these very small ones with genomic data. Our snail blot also shows we have more contigs, but still we get an N50 of almost 4 MB. For this tiny organism, it's really amazing. We have a Busco spore completeness 81, but we don't think that it does not indicate that we didn't sequence the whole genome. It's probably biological reasons that it doesn't have all metazone scores. This is not unknown from invertebrates. Now I want to switch a little bit gear, because when you work with invertebrates, what you really soon run into is that you run into cryptic species, that you don't know what these species actually are morphologically. And we had discussed at the museum a lot about it, and actually cryptic species is not just, it's more than a taxonomic problem, and I will come to this. But first of all, we need to think that differently about cryptic species, and we suggested a different definition where we split the taxonomic process of a species delimitation from the assignment of cryptic species. So first you do the taxonomic assignment that it's a distinguishable species, and then you look if they are phenotypically so similar that you wouldn't expect that. And then you assign the term cryptic species. One thing I want to mention here, which we found out and which I find really explained, because it's all about morphology with cryptic species, they are so similar. There was a surprising lack of morphological studies in all these studies, investigations. They didn't look at the morphology at all. And I mean, that's, I find that embarrassing. Pfizer et al. Came in the same year out with a paper where they looked a little bit what it means for ecosystem if we have different levels of species recognition. And they showed that it has an impact. This is shown here in the modeling picture. And interestingly, in the same paper, they came forward with a relatively similar new definition of cryptic species than we did. So maybe we did not everything wrong then there in our new definition. Jose Cerca, who was a former PhD student of mine, we took that up with cryptic species and looked a little bit more detail. This is an annelid, Steuercryptosepranamea, that was supposed to be one cosmopolitan species. We worked on the taxonomy, and we found actually it's 12 species, and the number of species is still growing. But we found only 4 morphotypes. Let me see, do we have a mouse here? We found only 4 different morphotypes, so that means some of these have identical morphologies. We cannot separate them morphologically. We looked a little bit more into these similar morphologies, and what we find is that the morphological disparity in Steuercryptosepranamea is extremely low in comparison to other annelid groups, which we use for comparison. It's about 8 to 10 times low for a very long time. And we dated the whole thing, and this morphotype 2, which would be this one here, together with this one, is unchanged for 140 million years. Another morphotype is unchanged for at least 65 million years, and one for at least 80 million years. So they stayed relatively similar, or identical, better said, for a long time. Actually, some of the morphotypes originated while this dinosaur still walked the earth. So they, since then, have not changed. We looked a little bit into the younger one of them, this morphotype 3, which is about at least 18 million years old. What could be the reason for that? And one was, okay, there might be still hybridization going on between them. With population genomic data, we found, okay, they are relatively, they still group in these groups as we find them, also using these kind of data, but we find shared ancestral signal. Then we tested this a little bit, or you say tested it mostly. Interesting, despite the shared signal, we did not find any indication of recent gene flow. So there's no ongoing hybridization between these different species. We also did not find any indication of bottlenecks. That was another hypothesis which was brought forward for their similarity. But we find ancient gene flow or incomplete lineage sorting because we could not separate these. So this seems to be something, this is old shared signal we see there, and it's maintained within the different species. What to make out of it is now the next step to find out. So this is still work in progress. I mean, you say finished, but hopefully someone will give out it. You see, and I also thought about that. What does it all mean with cryptic species? As I said, it's more than a taxonomic problem. We can use cryptic species quite a lot. So this is a taxonomic assignment of the different species. But cryptic species can tell us much more. As I showed you with the Pfizer paper, there is a possibility to understand a lot of ecosystem when we integrate cryptic species. However, as I showed you now with Stygocapitella, for example, we can also use it to understand something about evolutionary processes, in this case, stasis. But it's also possible for convergence. And then we can also use this, bring these together to understand then what impact do these have then, a better understanding of the ecological process going on for this. And there, we have a stable species, and this is where it occurs. It occurs at beaches, so it's nice to collect. I get to go to strands quite a lot and dig holes there. This is supposed to be a very stable ecosystem. It's erratic at a daily basis, but supposed to be on Earth unchanged for hundreds of millions of years. So we have a stable species, stable ecosystem. So maybe these can tell us something now with climate change and all the changes we see with mass extinctions. Stygocapitella, some of the morphotypes we see, survived mass extinctions. They survived a lot of climate change. If you look back 200 million years, there has been enormous changes in temperature over that time period. They survived it. So maybe when looking at these, we can learn something, what it means to be successful in changing times. How can I sustain this? How can I make it through to the next level when hopefully everything gets better? And with this having in mind, we are now establishing a hub at the museum where we want to look more addressing questions such as stability and discontinuity. This is called STADIS to reflect these two stages, and it's about stability in time but also in space. And the idea is understanding the properties and processes governing stability and discontinuity of biological systems at different levels of organization, from the genome to the ecosystem. So not just looking what I like, stasis, morphological stasis, but looking broadly. If you have questions to that, he's not here, he's in the moment in Brazil, but Johansson, you can ask, or me, because we are in the moment leading, we'll lead it, and it will start also next month in September. And with that, there were a lot of people involved through all this work and shaping the ideas. I cannot mention them all. I mentioned a few on the slides. Also, funding agencies contributed quite a lot. And you, now for your attention.
Thank you, Torsten, for these insights into this cryptic species and their genomics. We have time for a few questions before lunch. Anyone?
Everybody's hungry.
We have one over there. Let's see.
Why do you say that the beach is a stable environment?
Well, it's a stable environment. It is not stable on a daily basis. It's changing on a daily basis regularly with tides coming in, so you have changes in salinity, but that's changing all the time. And also, if it's rain shower, you get a lot of freshwater impact on a beach. For example, if algae gets put on the beach, you get a lot of food intake at that time. There's a lot of nutrition coming in. However, these conditions have been the same now and 400 million years ago. maybe not with large algae, but I don't know if they were already there, but at least 300 million years ago, there were large brown algae which could dump on the beach. You had rain, you had tidal actions already. So it's an erratic environment, but it's erratic for hundreds of millions of years. So in that way, it's stable. Even so, now, right now, it's not a stable environment, but over long term, short term, unstable, long term, stable.
Okay. Anyone else? Everyone hungry? All right. So thank you again, Torsten. Thank you. And
my name is Tore Slagsvold. I represent the owner of the journal, the Academy. I have been working on museums, and I have used museums collections, for instance, to look at morphology, the size of males versus females in birds of prey. Other friends of mine have been measuring eggs, shell thickness in raptors to compare across centuries to see if they got smaller or bigger, and also studied pesticides in feathers in collections of birds. But now we will learn more about how it can be used to look at genetics. So you can see here Niklas, he will then give us an insight on how that can be done. Please.
Is it on now? Now it's on, yeah. Yes, thank you. It's a great pleasure to be here. Yeah, so when I was asked to talk, I was asked to talk about the potential of museum collections to understand ecosystem function. But I am a systematist, and I mainly work with molecular data. I don't really know much about ecosystem function, so I asked, could I change my title to understand genetic diversity from museum collection, which is also what I do. But as I got into the subject, because I wanted to somehow connect this to ecosystem functions, I looked into papers that I had sort of seen flashing by in Twitter or wherever, where people had used museum collections to look at pollination biology or whatever. And I realized that museum collections have been used in many, many different ways. And I will introduce those ways to you before I get into how we can use museum collections for understanding genetic diversity. Before I do that, maybe I can just introduce myself. So I'm a professor in biological systematics at Lund University, but I'm also the director of the Biological Museum in Lund, which is, in fact, the second oldest natural history museum in the world after Paris. It was originated by Kilian Stobaeus, who was a teacher of Linné, back in 1735, and we are still around and have fantastic collections. I also want to acknowledge that many of the slides I'm using have been adapted from a talk given by Veronika Rota, who is our curator of entomology at the Biological Museum. So if we look at how many natural history museums there are in the world, there aren't a huge amount. There's a list on Wikipedia. It's not quite correct. There are clearly some museums missing from this list, but it gives us a general idea. So there's about 780 museums listed on this page, Wikipedia page, and strikingly, most of these museums are in Europe and North America. For instance, in Africa, there are listed only 32, and Africa is a hotspot of diversity. In Asia, 120. Whereas in the Nordic countries, Denmark, Norway, Sweden, Finland, and Iceland, there are 26 museums. So in such a small area of the world, we have almost as many as in the whole of Africa. And this is a problem, of course, because most of the diversity is not up here in the Nordics, even though we are happy with what we have. The largest natural history museums are also, of course, situated in the Western countries. We have London with an estimated 80 million specimens, the Smithsonian Institute in Washington, D.C. with an estimated 146 million specimens, Paris with 70 million specimens. And these are, of course, only estimates, because we cannot count. In Lund, we estimate that we have some 15 million specimens, or maybe 10 million specimens, you know, plus or minus 5 million. We know we have about 7 million specimens we have counted. We know we have 7 million, but there's a lot that we haven't counted. It's a huge amount of data. Yeah, the important thing to understand is that a lot of that data are concentrated in Europe for historical reasons. So the European natural science collections or natural history collections are estimated to hold about 1.5 billion specimens and hold about 55% of all the collections in the world, and hold about 80% of the world's bio and geodiversity. So we're also including the geological collections here with paleontology and so on. There is a big push now to unite all the data in European collections, which Veronika mentioned, DISCO, which is an initiative to bring together all of the European, mainly EU, but also I know that Norway is also involved, and UK, of course, and Switzerland, to bring together these museums and make the data available from a common portal from these museums, so that if you are interested in some species and you want to know where you can find these specimens, you can go to one portal and be able to find which museums have specimens of the species or group of species that you're interested in, without having to contact each of the museums separately to find out what they have. So before I get into the genetics, let's think a little bit about how biological collections are used. It begins with identification, and it begins with the type specimen. So when we describe a species, we assign a type specimen to that name, and if you are working on an obscure group of insects, for instance, and you collect a number of individuals, and perhaps there are no field guides available for that group or anything like that, then in some cases you actually have to compare the specimens you collected with the type specimens to be sure that you have identified it correctly. And where do you find those type specimens? In the collections, of course. And most of our museums are making available also reference collections, so the species which are around in the region. For instance, in Lund, at the Biological Museum, we have a reference collection for insects and plants of the Skåne region, so that people who are doing research can come and use our reference collection to identify whatever insects or plants or whatever they are working on. And this is extremely important if you're thinking about ecosystem function. You have to know what's in the ecosystem before you can study how they interact with each other and how they maintain the functions of ecosystems. So identification matters in many fields. Ecological studies are pointless if you're just talking about species 1 and species 2. You really need to know what these species are. With pest species, invasive species, introduced species, so somebody needs to be able to tell you if a ship comes into the port bringing wooden furniture from China and there's a strange beetle there. Somebody has to be able to tell you, is this beetle something to be worried about that might come in and become an invasive species, a pest species, and so on. And of course, disease vectors these days are becoming more, well, have been for a long time, very important to be able to identify correctly. Here's just an example of the fall armyworm, Spodoptera frugiperda, which in 2016 was first recorded in Africa, and within 6 years now it's causing multi-million, even billion-dollar losses in agriculture. Somebody had to identify that, and fortunately they were able to identify it very quickly, but that hasn't stopped the fall armyworm from marching across the whole African continent, and now has moved on to Asia, originally from South America. What Veronika and Hugo were talking about is a specimen is also data. It's a historical occurrence record, and we can do a lot with this kind of information. We can track changes in phenology. People have looked at herbarium sheets to see when certain plants have started flowering in the spring, and have shown that these spring flowers tend to flower earlier these days than 100 years ago. We can look at changes in morphology over time, changes in distribution over time, even atmospheric pollution, and I'll give some examples of these in a moment. So, my feeling is that the importance of historical collections is on the rise, at least in the eyes of biologists. And that's great, because for a long time, museums were thought to be just kind of forgotten places with collections where the specimens are collecting dust and nobody's using them. That's a very wrong impression, which is slowly changing among biologists, but we also want to change this, this impression with the general public and the policymakers. So there are challenges to using historical collections for assessing the loss of biodiversity or the crisis we're going through. And the first challenge is to ensure correct identification, which means we need experts. And this is what Artsdatabanken is doing here in Norway, training such people, and Artsdatabanken in Sweden also. But we also need jobs, as somebody pointed out, for those people who are being trained to be taxonomists and experts in identification. We also need to determine the correct coordinates if we want to use the historical information about where something was collected. We do need to know where that place was. I mean, if there's a label that says Lund on it from 100 years ago, in Sweden there are many places called Lund, so which one is it? So you have to know the history of the collector, where was that person at that time period, maybe read through diaries and so on before you can give the correct coordinates for a specimen. And of course, all of this data has to be made available. We've already heard about the FAIR principle, which is what I believe all museums these days are now strongly behind. So using public databases like GBIF and other such possibilities. Here's an example of how museum collections have been used to look at changes in morphology over time. So this is a very recent paper, just came out last week, where they looked at the asymmetry in bumblebee wings in collections and showed that bumblebees have become more asymmetric in the past 100 years or so. And this is most likely because they're more stressed today than they were before. So habitat changes, habitat destruction, climate change, all of these things are affecting how the bumblebees themselves are developing from egg to adult. And that information is there in the collections for us to get and to understand and to take forward to the general public and the policymakers. That you know, if we lose the pollinators, we are in trouble. Here's an example of how museum data is used also. This is just a very recent example. There are many such papers that have been published where people look at the ranges, distributions of species 100 years ago and compare them to where these species are found today. And the thing to note is that, no, I'm having the same problems as, nope. No, I used the mouse. Yeah, so the thing to note is that for this particular paper, the data was downloaded from GBIF, and I think it's fine. It's fine? Yeah. Yeah. So the data here is from museum collections. Even though it was downloaded from GBIF, the data from before 2000, that's all museum collection data, which has been digitized and made available through GBIF. Most of the data here is, whereas here we're starting to see the influence of these iNaturalist apps and other observation platforms where you can publish observations. But there are also, of course, museum specimens included in this. But what these are clearly showing is that species are moving. Their distributions are changing because of changes in the climate and the local environments. Here's a very nice example of how specimens were used in a way that the original collectors probably didn't imagine that they could be. So the paper was published some years ago looking at bird skins and measuring the amount of carbon, carbon in their feathers, black carbon, and they showed that that from 1800s to about, I think it was the 1940s, 1950s or so, the amount of carbon in the feathers of birds was increasing, and after that it started decreasing because there was a lot of noise about pollution, and there were policy changes so that one had to restrict the amount of smoke coming out of factories and so on. But what this paper then tries to say is that, you know, when we're doing the modeling of climate change, we are not actually taking into account the amount of black carbon in the atmosphere prior to the 50s. And maybe we should take that into account. Maybe that's explaining why things are happening faster than we expected with climate change. Biological collections are also important as voucher deposition, for voucher deposition. So when you are doing an ecological study, maybe you should keep vouchers of the species that you think you're studying, because, for instance, the methods that Torsten talked about, maybe there will be cryptic species discovered in the future, and then you don't know which one you were studying, unless there are vouchers available that you can go into. Anopheles mosquitoes, very important malaria vectors, are a good example of this. So the Anopheles species have been split into a multitude of species, and not all of them are vectors of malaria to humans. So if you want to know 50 years ago which Anopheles was flying around in a place where there was malaria or was not malaria, you have to go hopefully to the collections to find out. The biological collections can also inform us of interactions with other species. And this is especially used by pollination biologists, because you can go and look at what pollen is found on the bumblebees or the wild bees or on moths or on butterflies and so on. You can still collect the pollen and try to identify which species of plants those insects were visiting. There are also, you can find mites, or nowadays with genomic methods, you can find which bacteria or which fungi and so on are associated with a species based on the individuals in collections. Here's a nice paper came out fairly recently where they showed that when they looked at wild bees, they could show that the number of Rosaceae species being pollinated had increased over time, whereas the number of Fabaceae species had decreased over time. And the reason for this was because of changing grazing practices in the Netherlands. And this actually led to a decline in the number of species, wild bee species in the Netherlands, for there were more specialists on Fabaceae before. Here's another example of a small moth species which was considered to be one species, but then was split into two based on host plant specificity. And by going to herbarium specimens, they could show that the Antispila petri, which was formerly considered to be part of Trechkeala, was actually a very local species feeding on an endemic species of Cornus, whereas Trechkeala was feeding on an introduced species of Cornus and had been expanding its distribution following this cornus, which is planted in gardens and so on. Biological collections are used for red listing and so on. This is where the data comes from. Where were species before, where they've been lost from, are they declining at the faster rates, etc. But we can also use, and now we start slowly getting into how we can use museum specimens for genetics. So we can use museum specimens for conservation, for looking at population genetics and so on within species. And here's a nice example from Plebeius argyrogonomon, a butterfly species which is now declared extinct in Sweden. But Anna Runemark at our department in Lund has been sequencing whole genomes of the Swedish extinct populations and trying to find who are the closest relatives of this species in close by in Europe, which could be a source of individuals to reintroduce it to Sweden. Of course, the newspaper here is asking who killed the Plebeius argyrogonomon. The answer is that we did through our habitat destruction and so on. But we can use museum specimens to try and figure out where we might find populations to replace the ones that we killed off. Biological collections are, of course, the only way we can study extinct species. They're extinct, they're no longer in the wild. We have to go somewhere, and that is the collections to study them. Here's a very nice paper where there was the Xerxes blue which went extinct in the 50s or 60s in the US, in California. For a long time there was a debate, is it a subspecies, is it a species on its own? But these guys then went in and sequenced the whole genome, and the whole genome of the putative sister species, and showed that the Xerxes blue is genetically very distinct and should be considered a species, which is now unfortunately extinct. So yes, this we can do by sequencing DNA from museum specimens. And this is something which is only just now starting to really take off. I mean, people have used museum specimens for genetic studies before, but it's always been very difficult, very hard. But because of the amazing technological advances we've had that Torsten mentioned, now we can sequence whole genomes. And so now there are papers coming out showing that we can extract DNA. There is DNA in museum specimens. And this has now been done quite a bit with insects. And here's just an example of mosquitoes. Here we have the specimen before DNA extraction. Here's the same specimen after DNA extraction. And this paper showed that it was a methods paper showing that if we use different kinds of buffers, we get different amounts of DNA out. And they gave a protocol which they recommended for how to get the best quality DNA out of old specimens. Here's another paper which came out just last week again, where basically they showed that the older you get, the smaller the fragments of DNA are in your specimen, and that's not wholly surprising. That's what we expect, that the DNA, as it degrades over time, it becomes more and more fragmented. How old can one go? As old as there are specimens, I think. The oldest that we have had success with was a specimen collected probably in the early 1800s. The label doesn't say, but it was a type specimen for a species described by Zetterstedt, which is kept in our museum. And we were able to, or Hamid Ganavi, along with Markus Stensmere, were able to extract DNA from this old type specimen, sequence it, and it mapped well to a reference genome of the species. So we can go back 200 years easily. I can say that the specimen, it did fall off the original pin, so we had to repin it, but in the process it got a bit cleaned up, maybe. We're still trying to figure out the best way to extract DNA in a non-destructive way. So here we still have something to develop so that we can get, so that the eyes don't collapse like it did in this individual. Anyway, museomics is something that I have been very excited about for the last 5 or 6 years. So we are doing a lot of whole genome sequencing from museum specimens, whole genome sequencing also from existing DNA extracts. So I've been doing PCR-based work for the past 20 years. I have thousands of DNA extracts. And now we can sequence the whole genomes from these very same extracts that we did our PCR-based work from. But what I wanted to share with you today was basically just a proof of concept. When we sequence museum specimens, are we getting the DNA of the specimen that we're interested in? So we did this was a PhD project of Elsa Kahl, and we asked the question for the very common species Pieris napi, how much butterfly DNA are we actually sequencing when we extract DNA from these museum specimens? And can we get a good coverage of the genome? So here are just some basic results. So here's the range of years that we had for the specimens collected, starting, the oldest one was from 1885, the newest one from 1989. The average size of the fragments in these DNA extracts was very small, and it decreases over time as expected. But yeah, so we're talking about fragments about 50 base pairs to 80 base pairs, or if it's a really good specimen, which we didn't have in this case, you might get 100, 120 base pair fragments of the whole genome. The nice thing was that these fragments did map to a reference genome very nicely. So about 80% of the DNA that we were sequencing was actually the butterfly's own DNA. What the other 20% is, well, there's some of it is interesting, like Wolbachia, which is an indigenous bacteria within the cells of the live butterfly, but some of it is not interesting. It's humans, it's whoever collected it or whoever did the extraction, or there can be fungus as well that has even invaded the specimen afterwards, and so on. The coverage decreases with time, so you don't get as much DNA from the older specimens as from the newer specimens. And so, yeah, but still, from the oldest specimen, we did still get around 7x coverage for the whole genome, which is, it's enough to be able to use that data for analyses. I'm not going to go into mitochondrial coverage is extremely good, but that's kind of expected. So we're at this stage, really, that we're still trying to figure out the best protocols, how to extract DNA, how to sequence the DNA, how to deal with the highly fragmented genome. So there haven't yet been very many studies where this has been used to actually look at how things have changed over time. But many of them are in the works. I know I've heard presentations. We also we're working on the Pieris napi data and so on. But there are a few papers that have been published. The first one here showed for 2 species of butterflies that over the past 100 years, they're both endangered species in Finland. The heterozygosity has decreased over the past 100 years in these, so they've become more homozygous, these butterflies. And for the top butterfly, Erebia embla, the isolation by distance has increased over time. So the remaining populations have become genetically more distinct from each other because of lack of gene flow and so on. A similar story from the other paper here, from the same issue of Molecular Ecology Resources, where they looked at museum specimens of a species of Cardamines in the Brassicaceae, where they showed, it was basically just a showcase of what you can do. And they showed from 2 populations which were several hundred kilometers apart in Germany, that the two populations are genetically more distinct today than they were in the past, mainly because the gene flow between these populations has been cut down over the years because of habitat fragmentation and so on. So the potential is there. There is a lot of potential in how we can use the genetic resources of museum collections. So I think museomics is definitely a way forward. As I pointed out in the beginning, natural history museums hold vast numbers of specimens going back over 2 centuries at the best. And so we can use these specimens to look at the population genomics of important key species, if you like, in ecosystems, both of rare, which is kind of the low-hanging fruit, but also of common species. So one of the things that I'm really keen to try to look at is with a common species like Pyrrhus napi, which is flying all over the place here and in Skåne and so on, are the individuals that are today flying in Skåne more genetically related to German Pyrrhus napi from 100 years ago, because climate change has allowed those phenotypes to move north? We don't see it because they look the same. We can see it with species boundaries moving, so a new species comes in. But what about the species which are very common? Are they genetically now more similar to southern populations from the past? We don't know. Nobody's looked at that. And of course, museum collections are the only way that we can look at genomics of extinct species, and there are all kinds of crazy ideas out there to bring back mammoths and thylacines and whatnot. But there are also serious things that we can do with looking at extinct species, looking at genomics of those species which didn't make it in this changing world. One might ask the question, okay, we have 1.5 billion specimens here in Europe. Do we need more? Because if we want more, then we need more space, because collections take up space. The short answer is yes, we are right now in an amazing period of change, amazing scientifically, disastrous socially, and we need to keep on collecting so that we have a record of what's happening right now out there, so that the future scientists can look back and say that, oh my God, in 2022, these species were here, now they're far away from here, for instance. So yes, I think it's crucial that we continue expanding our collections and getting more specimens into them, so that we have that record of what's happening today. So yeah, museum collections are critical to understanding the future. We need to understand the past to understand the future, and the museum collections are a window on the past. And in my opinion, the uses of the museum collections are basically limited by our imagination. So we are now using collections in ways that nobody even thought about 10 years ago, 20 years ago, 30 years ago. So yeah, that's what I wanted to tell you about museum collections, and thank you for your attention, and thanks to my colleagues for help with various things.
Thank you, Niklas. It was a very good overview, and I'm kind of regretting that I left the museum. Some questions. We are a little bit late, but I can take a few. Down there.
Yeah.
You said that many of the large museums don't have, you have estimates of how many specimens you have. How are the efforts of actually figuring out what the museums have, and what can be gained by actually figuring that out?
So we want to, of course, digitize. This is the big thing now. Digitize all of the specimens. And for some groups of organisms, this goes relatively quickly, like with plants, which are on flat two-dimensional pieces of paper that can be scanned in. You can have OCR reading the labeled text and so on, and they are relatively easy to digitize and count. But then you have insects which have labels. Sometimes there can be, 10 labels, one under each other on a very small pin. There might be 10 insects on one pin. There are all kinds of difficulties there with the digitization. The main point with that is that if we have 146 million specimens, how long will it take to get the information? All of that information is basically locked in the collection drawer until somebody digitizes it and makes it available. How long will it take to do 146 million specimens? It would take more than 146 years for 146 people to do it. So it's, that's why we want to know how much is there, what's the challenge ahead. And for us in Lund, where we know we have 4 million insects, but we have only digitized about 200,000. So that, and that's taken 10 years to do 200,000 with the resources that we have.
May I have a little question there myself?
Yeah.
I have been collecting insects a lot some years ago. I was so impressed by how you could extract the DNA from this dry little Drosophila of yours. Can you say a little bit about the technique, and is it very time consuming?
No, so the extraction is not very, it's a normal DNA extract. There is DNA in most of these specimens, not all. So we have occasionally complete blanks where there's no DNA left. And occasionally we get lots of DNA, but it's not the specimen's DNA, it's fungus, which has invaded the specimen afterwards and has completely eaten up the specimen's own DNA. But the vast majority, about more than 90%, we get the DNA of the specimen. And it's really the sequencing part that takes longest, but the extraction, if there's DNA, it's easy to extract from there. And
you had a question, Lise?
Yeah, you had a very nice plea for further collecting material and store it in the collection. You also mentioned that we have use for these material later on that we could never imagine earlier. Would you change or forward recommendations? How should we and what should we collect today, given that we see unprecedented opportunities in the future? So should the collection practice change because we see future potential?
Of course, nowadays people are collecting for specifically for DNA, so collecting into cryobanks and so on, but that's very expensive. For instance, in Lund, we have no possibility to have a cryobank. So we can't change that, even if we would want to. On the other hand, storing specimens dry or in ethanol or whatever, that's possible, and that seems to be good enough for most cases. Whether we should focus on some species groups, I don't think that's a good idea, because again, it limits our imagination. Then if we don't have all of those weird little insects that nobody knows anything about today, if they're not in the collections, then we can't go back and look at them. So I think we should continue collecting broadly as much as possible, and just making sure that they are properly curated, findable, and in that way they will be studied then someday. Because if they're just collected and pushed into a drawer and nobody even knows they're there, then we can't use them.
I had a quick question. So with the collections that we have today, we can study things that we never imagined that we wanted to study in the past when we collected them. And there's a strong case to collect more material today because we'll have questions in the future. But in both cases, we are studying things that happened in the past because these are actually collections. So back to your statement, is like it's critical that we collect to understand the future. So then we're talking about the predictive ability of research. And that is always something that I really struggle with in making a strong argument for that.
Well, think about distributions. So over the past 100 years, we've seen that species they're not going to stop moving today. Yeah. So we've seen that they will continue moving. Yeah. Why, where, how? Okay, we can use the past data to understand what factors have allowed them to go to where they are today. and then predict the future based on that, for instance.
Yeah, but then we still need to do something with that information.
Yeah, absolutely. No, I mean, I'm not advocating for
I'm always looking for real strong cases that illustrate this.
For the policymakers, when the pest species start moving up and coming here, and we want to know when they're going to arrive in Sweden or Norway and start eating our crops, that's pretty important information.
Yes, in a way we should put our heads together and write more review papers that talk about that, understanding the future potential.
Yeah, for sure.
Thank you.
Yeah, thank you.
Okay, thank you. Then we have to go on.
Yes. Okay, thank you very much for inviting me to this symposium. Although I'm not a taxonomist, I never worked in phylogenetics, I never described any new species. I'm a marine ecologist by training, and I'm now at the Alfred Wegener Institute for Polar and Marine Research in Bremerhaven, and I had there a working group in the Department of Functional Ecology, and the name is Ecosystem Functions. But we do biodiversity research in polar waters, but not only polar waters. And our intention is, or our approach is, to work data-driven river. We started to do local studies, but we discovered that we have to upscale our results to larger areas to come to more common or general conclusions. And then we changed our approach. And we work with data sets, and we discovered that at least in the marine realm, it was difficult to get the data from polar waters. And we faced a challenge, and we had a solution for that, and we developed a operational information system which we called CRITTERBASE as a solution of the fair biodiversity data challenge in polar waters. It's not only me, of course, who worked on that, but several co-workers in the Alfred Wegener Institute and in other institutions. What do we do? A little bit of background information. What we discussed it. What we do is functional biogeography. So we work in a spatial and also temporal context. And our mission is to understand the interactions between taxonomic, but mostly functional and trophic diversity in the significance for ecosystem functions in a spatial, temporal, and also anthropogenic, climate-related context. We look at different biotas, both on organism level, but also mainly on community level. We look at the environment, human impacts, some ecosystem functions, not all of them, of course, and they are very often related to some goods and services that our funders are interested in. So that is the overall, the common approach, so to say. We do that in different regions. At the Alfred Wegener Institute, for instance, on Arctic ecosystems, which are, as all you know, under environmental pressure, maybe more than other regions in the world. And we also do it in the North Sea, because this is the waters very close to Bremerhaven, of course, but also in the Antarctic. And they're mainly in the Weddell Sea area, which is over here. In all these projects, of course, the environmental change that is going on is let's say, the main intention to give a baseline and to come out in the end with predictions of the range difference in the future. And how do we do that? What is our common approach? We are data-driven. We build an information system. We also have metadata that we are working on, so it's can be called knowledge systems. We get the ecology right, first of all. We define our needs, parameters, scales. We collect the data, fill missing data, curate the data, which is much more challenging than I thought it is before. Then we turn the data into information. We secure the data curation, at least in our institute and in a larger context. decoupling to external databases, and also environmental models, which are developed not only in our institute, but also in the institute. In the end, these data are modeled and analyzed to create more scientific knowledge on the system functions, and we help also the modelers to look at future development of ecosystems or biotas in polar waters and also the North Sea. And the Alfred Wegener Institute is a Helmholtz Center, so it's always also has an applied perspective, so to say. We produce some outreach products for some other stakeholders, mainly decision makers. Okay, so we have different data types, so to say. Most important, of course, are the biotas, which are species or other biotic data, but also environmental data, functional traits, and data on trophical relationships. We have a strong GIS department, which can do that in a spatial context. If that's available, then we do some modeling with the help of other departments, both on an organism level, but also if you relate that to the scenarios of different climate developments in coupling models to climate. And for instance, one of our products in scientific knowledge or outreach is that a new marine protected area is or should be established in the Antarctic, in the Weddell Sea, and in our institute, the pilot project has been founded to do the information compilation that prepares the proposal of this marine protected area in the Antarctic. Good. What is our data-driven research? So in our first step, we look mainly at ecological BIF, for instance, but others are not. And in the Antarctic, it's mainly our collaboration with the British Antarctic Survey, also Norwegian partners and international projects. So data is available, and some of these data is also available from repositories, quite a lot of repositories, like IPBES, for instance, GBIF, the Polar Data Catalog, and others, which are given here, just as examples. But these are all data repositories or portals to metadata. So these are not operational information systems that can be used actively. And so we decided we have to do it on ourselves. We have an extreme multidimensional data diversity in sampling devices, methods. They are very different, they are very diverse. Even in sample analyses, different parameters at different resolutions have been sampled and so on. And this has direct consequences, of course. There is lack of so-called collective data knowledge, so to say. The merging of data is difficult, very elaborate, sometimes even impossible. And some data are, at least in polar research, simply lost. And that if we feel, in that sense, not we, in general, feel the big data intention to use big data sets to come up with more general conclusions on first principles. The biodiversity ecological complexity relationship is difficult to study. And also the coupling between ecological or ecology or biotas, so to say, and the environment. And also for the stakeholder obligations. So the data heritage was not guaranteed, at least in a polar environment, so to say. And the solution was to come up with an operational data environment that we can use for the exploration of the data, of organism-related data, which can handle different sampling devices, methods, integrate all types of possible sample analysis to comply with the FAIR principles, which have already been mentioned in other lectures. And this is a name CRITTERBASE. It's only marine critter at the moment, and mainly benthic, so seabed organisms, which are in that database. And we have 3 different regional components, so to say. One is called PanAbio, Pan-Arctic information system on benthic biodors, which can also be upscales in the long run on even including zooplankton or others, marine biodors. Bionosis is the benthic North Sea information system, which is a component of CRITTERBASE. And then we have the Weddell Sea, the Antarctic one, which is also included in that at the moment. So what is the actual state of our efforts, the current status? We published a paper which came out recently, 2 weeks ago, so to say, in Scientific Data on CRITTERBASE, a science-driven data warehouse for marine biodor, which can be downloaded using this DOI. And it describes the components and the opportunities of this thing. There are different components. One is the data space with the working platform, the data model, so to say. The working platform is an open portal, which has been launched now for polar researchers, but also for the general public, which can be accessed using this URL. And CRITTERBASE is described here, and it has different components. One is the mapping and download component of the data that is available. And this is just an example of one of the user interfaces for the download on the mapping, and you can apply different filters on that. For instance, region-wise, sorted according to the FAO, Food and Agriculture Organization, the regions, and these are the different data from the Antarctic, for instance, or here in the Northern Hemisphere from the Arctic and the North Sea. And then that can be downloaded and used for further exploration or data analyses. This is the current state of the areas. We have 3 at the moment. The sampling methods that are represented in this information system. And these maps give an overview about the spatial coverage of the data that at the moment are included there. And you see most of them is here in the Arctic realm. Then we have put a lot of efforts into the data model to have it as comprehensive as possible. It's modular and can be extended relatively easily. It looks like that. So a lot of metadata is included in that, and also some biological information that can be included if it is available. A big problem was the taxonomy, of course. You all know that maybe even better than me, because names are changing and also the phylogenetics, and we decided to not do it at ourselves, but we have a continuous update and a link, so to say, to the World Register of Marine Species, so that we always rely to the APIDs that are included in this worldwide, in the global marine database. And then we created a collector app, which is a possibility to have old data, at least in the polar research. Many data are stored still in Excel files from expeditions, for instance, and to put these into the data we created in our data system, we created such an app, which is an SQL app, which is relatively easy to use to collect data from other sources into our system. And on the other hand, we have interfaces to data archives like Pangea, also through IPBES, which is part of GBIF, so to say, so that we also can save our data in to other platforms, which are even wider in coverage than our operational database. This is just the beginning, of course. The perspective one is, it should include a kind of, or it is a showcase of a national biodiversity data initiative in Germany. There are several use cases, and CRITTERBASE is just one example of that, focusing on marine and polar data. And then we are working at the moment at an analysis app so that we can do some basic data exploration. And this is, hopefully that it works. It is a quick time movie. Here we have an R script that extracts some data from the database, from CRITTERBASE, and relates it to some environmental data. And in this case, it is Brantiostroma, which is a North Sea benthic animal, and we relate it here to the medium sediment grain size. And you see that there is some relationship for this species, just as an example of what we are working on at the moment. Good. In the long run, we will relate our database to other databases which are currently developed in the Alfred Wegener Institute and in some other institutes. For instance, we will look, that's the current status at the moment with the relationships to other databases. Then we will work on not only the taxa, but also in the long run on the traits, link it to environmental data, and do spatial distribution models for some key species, at least. And these are interrelated through this central hub, so to say. One thing is, the first thing that we are working on at the moment is to relate our taxonomy data to traits. There's an Arctic traits database, which has been initiated by a former PhD student, worked at the Alfred Wegener Institute, so that we can also do the analysis not only on a taxonomy basis, but also on the functional traits basis. And in terms of the environment, we have our own environmental database, which is Pangea, but we also related it successfully, for instance, to the Google Earth Engine or Bioracle or some other databases that are known here. And through the finite or FISM, finite element sea ice ocean model that has been developed in Germany, we can relate our spatial distribution models to the future or the possible future and come up with some, let's say, information that can be used by IPCC assessments, for instance. So it's interesting for some partners and stakeholders, both in science and in an outreach or stakeholder context. For ourselves, of course, our partners in Germany, especially Pangea, which is a great environmental data information system that is very much used in Germany. In an Arctic context, we work closely together with the Arctic Council Working Group CBMP, which is part of CAF, of the Convention of Arctic Flora and Fauna. And in the Antarctic, as I already told you, we are involved in the, let's say, project that is preparing the proposal for an Antarctic MPA, Marine Protected Area, in the Weddell Sea. Here, SCAR. Yes, and then we have a running project, COASTCARP, looking at the carbon fluxes as an ecosystem function in the Antarctic, which is part of the Horizon 2020. And also with IPCC, we have some interrelationships. So thank you very much for your attention. And it's just 20 minutes, so there's some time for questions left. Thank you very much.
Thank you very much, Dieter. Only to tease you, but I thought a crab had 10 feet, like a decade.
Yeah, it's not mine. We had another logo, and this is the logo that has been prepared from our public relations department. Okay. But we will. Our logo was not good enough. Okay. Which was a fish.
Well, any questions?
Yeah, I have a question about this data in CRITTERBASE. You essentially rely on data on biodiversity or species observations that others have done. Can you give
Not exclusively, by the way. We also sample our own data, but it's only a small part, of course.
Okay, but can you give a rough estimate? What would you think, how representative is CRITTERBASE in your 3 target areas?
I mean, it's most representative in the Arctic, because we inherited, so to see, the legacy of the Arctic Ocean Diversity Database, which was established in the framework of the Census of Marine Life, and then the project was over, and it was not funded anymore. They also had a portal, and a lot of Russian data was also involved or included in the Arctic Ocean Diversity Database. So if you remember maybe the map, there's a lot of Russian data, mainly from St. Petersburg, from the Zoological Institute, St. Petersburg, of the Russian Academy of Sciences. And in that regard, at least it's only the benthos at the moment. It's just benthic animals and some plants too. And in that regard, we are quite, at the moment, it's currently, we hope it will increase, of course, in content. In the Arctic, it's quite, it's not complete, but the coverage is quite good. In the Antarctic, it's at the moment only the Weddell Sea. So it's not circumpolar.
Okay, more questions?
And the reason for it is because we work on this Weddell Sea marine protected area. We sampled all the data which are available from that region, because that's the target region for the MPA proposal. And most of European research, by the way, is taking place there. The Americans look more on the Ross Sea, and we are in the process of making, let's say, a deal with them. But the Europeans, the Germans, the Norwegians, and the British are mainly working in the Weddell Sea area, because it's the Atlantic sector. It's not that remote for us to go there.
You mentioned that you're working with traits, functional traits. I'm very interested in functional traits. Can you say a little bit more about it?
Yeah, functional traits, there's a lot of discussion, of course, at the moment, and for that reason, I was not involved in that. Our PhD student, she was very interested in that. Now she moved to Vienna. She's an Austrian, she's now gone back, and she initiated this portal on the Arctic Trait Database. And we look at different types of categories and different modes of these traits, and they applied the fuzzy approach, so that one organism or species can have different modes in the category, which can be trophic or behavior or the body size or others, the usual ones, the usual categories that are used, at least in zoology, in marine zoology. But there is no, let's say, no commonly accepted, at least not in marine, commonly accepted comprehensive scheme of that. They are very similar in the names that the categories that they use, but there is no standard. So we decided to use this Arctic Trait Database, which is an international initiative, a pan-Arctic initiative.
Okay, it's time for coffee, but I will give you a big hand again.
Thank you.
Thank you.
Okay, I think we're all back now. So it's time for the 2 final talks, which both are about metabarcoding, which has become a very useful technique for assessing biodiversity, especially, I think, in connection with eDNA. I'm not sure if you can talk about eDNA, but it's... No. Okay, yeah, anyway, so it's the first talk is by Paola Papalardo. She's a senior postdoctoral researcher at the National Museum of Natural History in Washington, but I think you're based in California.
Momentarily.
Okay, so please.
Good afternoon, everyone. even though I'm going to talk about the role of taxonomic expertise, be warned that I'm an ecologist. So it's my perspective as an ecologist. I do study biodiversity, and assigning taxon names is very important for biodiversity research and conservation. And the traditional way of assigning a name is to do a careful morphological assignment to identify an organism, hopefully 2 species. In some environments, such as the midwater, where we have tiny organisms, high diversity, this may be a little bit more challenging. And there are advantages and disadvantages. We can, for example, assess sex and reproductive stage if we use morphology. But we do require high taxonomic expertise, and sometimes it's not possible depending on the state of the specimen, or if we have early life stage. And more importantly, it is time-consuming. And as has been mentioned before by Hugo, in this current biodiversity crisis, we are maybe losing species faster than we can describe them or that we can understand their role in the ecosystem. So a faster way for species identification that has come up is barcoding. So same way that we identify products in the grocery store, we could identify an unknown sample by extracting DNA for a small portion of DNA, and after comparing it with a reference database of known sequences, we could be able to, if we have a good match, to assign a name for that. There has been here debates between species identification using morphology or barcoding, which I'm not going to get into. I want to highlight the advantages of barcoding that can be cost-effective and fast. It also provides useful phylogenetic data for a lot of studies and can identify cryptic diversity. And I do want to highlight that they are really interdependent, and has been mentioned before in other talks. We do need a very accurate identification and a quality sequence to be included in these reference databases. And then now, with the advance of the genetic technology, we can extract DNA from a mixed sample of organisms. We call it metabarcoding if it's like, for example, from a plankton toe or from a net, from a trap, and eDNA when it's a sample from the environment. And then we can have multiple sequences at the same time that are compared to this reference database, and at once we can have a list of all the organisms that are present in the sample. Now, you might be wondering if this works. It does work. It varies between the taxon that you are studying and also sometimes the environment. It can range, for example, in studies that of mock communities where the composition is known, it can vary the recovery between 40 to 100%. When we go to study real communities, especially in the marine environment, you still have sometimes large percentage of unknown samples that you cannot identify at all. There are some tools that are coming up to try to reduce these numbers. For example, the dark matter investigator tool. So it's getting better, I think. Now, metabarcoding involves multiple decisions, from the type of genetic marker we use, the bioinformatic pipelines, how we cluster those sequences, the choice of reference database, and the choice of method for taxonomic assignment. And unfortunately, there is not much consensus on method. You may go through a rabbit hole if you try to pick one. A recent review by CRITTERBASE and collaborators show a lack of bioinformatic standardization and also poor description sometimes of the bioinformatic method, so it's also hard to understand what is going on. So today, I'm going to be focusing on this last part of the workflow from the reference database and how we assign taxonomy. And I'm going to be talking about what I think are 3 things that could help to improve taxonomic assignment. One is to develop a local reference database. The other to help curating and expanding public reference databases. And the last one to use optimized thresholds. So for the first one, I'm going to present data from the Smithsonian Stream Code Collaborative Project that was assessing zooplankton biodiversity in the Gulf Stream. And basically what we asked was if a local database could help us to improve the outcome of metabarcoding. And we have this very simple plan to collect zooplankton samples, to analyze them using morphology and metabarcoding, to use those samples to develop a local barcode database. And by local, I mean geographically local. And to compare the metabarcoding results when using public databases or using our local database. So in a relatively short time frame, like 2-week sampling, about 20 people, experts in different taxonomic groups, we collected 1,500 samples and about 2,100 barcodes for 2 genetic markers, 18S and CO1. And we wanted to check what was the novel contributions in these barcodes. So here you can see the novel sequences for each marker at different taxonomic levels. And for the order, those may be a little bit overestimating because of taxonomic discrepancies. But overall, you can see that in this short time frame, we contributed a lot of novel sequences. Then we also analyzed the same zooplankton sample with metabarcoding. And you remember that simple plan? Well, when we add the metabarcoding part, this is more like the reality and what you could expect on a metabarcoding project. There are, as I mentioned before, multiple variables. We have 2 genetic markers, different type of clustering, and different reference databases and methods for taxonomic assignment. I'm going to show you some data for CO1, for OTUs, and for the RDP classifier using Midori, and for BLAST-N using GenBank and using the local database, which is the Streamcode database. So here you can see the community composition for CO1 represented as the proportion of OTUs and for the 3 methods to assign taxonomy. You can see that the majority of the samples were unidentified, which is something common, especially for this marker. And after that, the largest representative were the arthropods. If we look at the samples without the unidentified and without the arthropods, we start seeing what are the other groups that are represented. And we did observe different representation with the local database. For example, look at the annelids. We observe more annelids. So I'm going to zoom into that group and show you the number of unique OTUs that was found with each approach to assign taxonomy. So this was with the RDP classifier and the Midori database. This was with BLAST-N and GenBank, and this with the local database. And you can see how each contributed with something different, but we have more success with the local database. These were samples identified by Karen Osborne, my advisor at the Smithsonian, and those were the specific contributions to GenBank for annelids. Now, this was only to phylum. If we go a little bit deeper or like lower resolution to family, here you can see the unique families for annelids. So these in green, where the families identified by morphology. These 3 families were identified both by morphology and also by metabarcoding using the public databases. Now, if we look at how these change when we add metabarcoding using the local database, we can see that the identification to family increased, the family detection increased, and only 4 of them were still only identified by the experts. So the take-home message from this is that a local database can improve the identification of unknown sequences. And then also if you publish this good quality sequence you created, and if they are associated with a voucher, that is also important, like we have in this project, then you're also able to help with point 2, which is to help curate and expanding public reference databases. There are a lot of databases, so it's hard to choose. They differ in many characteristics, some of them the type of genetic marker that they cover, or taxonomic group, also how frequently they are updated. And you may wonder if these differences are important. Well, I'll show you some data of our student, Anish Araday, that look at the performance of different CO1 databases. Here you can see some of these databases for 2 taxonomic groups represented in the midwater, Hyperis and Theropods. And I'm going to show the % of correct assignments to different taxonomic levels. So you don't need to worry about the details. What matters here is that the accuracy of the correct assignments vary between the databases and also the taxonomic groups. An example from the work of Robinson and collaborators for bacterial and archaeal community composition. In this case, they compare 4 reference databases and they had 15 samples. So if you look across databases, the colors represent family level taxonomic composition. You can see again that for most samples, you can recover different composition with the different databases. So they can be different. And you may be wondering, well, could it be because of errors? What is, are there errors in these databases? Some papers present a large amount of errors, such as 10 to 21%, for example, for fungi. There are other papers with a little more positive scenario. Lurate and collaborators found between 0.01 for phylum assignments to 3.5 for genus, studying many mitochondrial sequences. And when they were able to determine which was the likely source of error, for example, for phylum, class, and order, they found that it was likely a laboratory contamination, whereas for family and genus, it was likely due to taxonomic revisions that were needed. Similarly, the work by Van der Borg found 6% of errors for amphibians for cytochrome B, and also they found that the large majority of those errors were for what they coded like wrong taxonomy, that is, they were initially incorrect or updated. And why I think this is positive is because the taxonomic errors can be fixed by the NCBI taxonomic team. And let me show you a couple of examples of that. This was a case where my collaborator, Alan Collins, flagged to NCBI a species name that was unaccepted, Beroe pileus. And the taxonomic team at NCBI fixed this to Pleuroblanthe pileus, the accepted name in worms, like within a week. And now, even if you search with the old or the new name, you can find the correct sequences with the correct name. Similarly, I found an error with wrong higher taxonomy for the taxa Alcyonium glomeratum that appears as a bryozoan, but it looked to me, based on a couple of things, that it was a coral. So I reported this. They look at it and was fixed also, and now it has the correct taxonomy. So basically, you also can help to improve GenBank. You can report updates to that address up there. Sometimes we, I say we for my taxonomic teams that I'm associated with, you can update your taxonomy, and it's important to update it in GenBank. You can also, it's important to check that if you have sequences in embargo, that they are released when your paper is released. And also you can report errors. You can suggest, you can use taxonomy question. That's what they suggest if you know that it's a taxonomy issue, or other sorts of errors that they can contact the submitter to fix them, or they can flag them and then they will not appear in a blast search, for example. And in our experience, these errors have been handled very quickly by the taxonomic team. There are other data curations efforts trying to improve these databases. For the 8th International Barcode of Life Conference here in Norway, they did a hackathon to create marine barcodes. And they, this team analyzed about 7,500 barcodes, classified them to see if they were correct or likely misidentified, etc., and flagging them for the, these were from BOL, for the BOL team to try to solve those issues. And sometimes it may be harder to get a group of researchers to spend so many hours in this. There are other efforts to do some kind of automated system, such as the work by Fontes and collaborator. They develop Bags, which is an R-Shiny application that will download data from Boer and grade the data by quality based on the beans, the genetic beans, that is how different they are genetically, and the congruence with the species name that they are assigned. And at least you could have an idea if you see something weird when you use them, if it's for because of some of these issues. There are also several working groups, and I just have a couple here, but there are also other initiatives that have been mentioned here today to look for best practices in barcoding or metabarcoding, or to generate new barcodes. And there are plenty of barcoding gaps out there. Here I plotted the percent of species that have CO1 sequences in GenBank, and I did this from the Midori database for different phylum. And you can see that there are a lot of them that have a lot of species that are not represented. Here, even for species that are used for ecosystem quality measurement, here a report by Wegan and collaborators. You can see, for example, as low as 22% of representation of these species in genetic databases for marine invertebrates, and the largest representation was for freshwater fish. And the last example is from the MetaZoo database that shows also how different representation can vary for a taxonomic group. So zooplankton, you have 25% of a species represented with barcodes, and ichthyoplankton, you have much more, like to 67%. So the take-home message is this is teamwork. We are all using these databases, and we can all help improve them. So try to keep your assignments updated in GenBank and report errors if you find them, and publish your sequences. The 2 projects I have been involved, we publish about 2,700 sequences. So the last tip is to use optimized thresholds. Here you can see a table of papers I compiled from marine and estuarine environments. This is not exhaustive, but just to represent that you can find different genetic markers being used, different reference databases, different methods for taxonomic assignment, and also different confidence thresholds used to trust on the taxonomic assignment, or to decide on an assignment. So some databases have recommended thresholds included. For example, here, Porter and Hagi Babe. In this case, this is to use with the RDP classifier. And if you want a 99% correct assignment, here you have 4 different taxonomic levels and different types of sequence legs, recommendation on what you should use. But you have to have in mind that these recommendations will be specific to the reference database, and also to the genetic marker, and to the method you use for taxonomic assignment. So hopefully you can look for them. They may be available. And if not, the recent reviews also suggest you need to try to optimize the parameters for taxonomic assignment and which of these confidence thresholds you use. One way of doing that is using mock communities where you know the composition, and try to tune that to optimize the recover what you know is there, as has been shown by HLeap and Bokelich and collaborators. Unfortunately, there is always a trade-off between accuracy and misclassification as we move in thresholds. So if you want to try to be very correct, you are going to leave a lot of things unclassified. That has been shown by Gold and collaborators. And lately, there are new tools that are being developed constantly. Here, an example of a tool that has been developed to optimize thresholds in fungal sequences, but it probably has a more widespread applications. That is, this one is called DNA barcoder. And myself, in my current project at the Smithsonian, with my collaborators up there, we are working on developing taxa-specific confidence threshold for different taxonomic levels and to improve taxonomic assignment. So we are doing some benchmarking with known sequences. I'm using the Midori database for this, and then testing these different thresholds with real data sets. And I'm using BLAST-N for taxonomic assignment. So I'm going to show you some data for CO1 again. My test sequences were from Midori longest. And I'm going to show you some matches to phylum for today and with the stream code data. And the reason why I chose BLAST, even though it has some critics, is because it is the tool more widely used. Here, from the review of CRITTERBASE and collaborators, are compiled the data used for taxonomic assignment, and you can see the number of papers that use each tool, and BLAST-N was BLAST in general was the more widely used. So let me show you how the number of sequences can affect classification. Here you can see the % of unassigned species for sequences belonging to different phylum as a function of the number of sequences. And here you can see the % of incorrect sequences as a function of the number of sequences. So for the phyla that have low representation in reference databases, there are many sequences that will remain or unassigned or that they will be assigned incorrectly. Conversely, the more sequence we have, the larger the % of correct assignment for this phylum. And then I set up to find the specific threshold for each phylum using an ROC framework. So an ROC curve plots the true positive rate as a function of the false positive rate. And this is the area of where a classifier does well, and this is the area where it becomes worse or it's not even better than a random classifier. Let me show you an example with some real data. This is for a mollusk. And as the confidence threshold here, I use the % identity. That is one of the more used and more intuitive. So as you move in the ROC curve, you go from an area of high thresholds to lower thresholds. And you can calculate the best threshold for the taxonomic assignment of mollusk with different methods. This one I made with the Uden method. And you can also measure the area under the curve. That will give you an idea of how good the classifier is. In this case, that is 0.91, which will be like a very good classifier. So with mollusk, if we use an 82% similarity as a threshold to decide this sequence is a mollusk, we can have a very good classification. So then what I did is I used this method to calculate the optimized threshold for each phylum. And then I compared that with using a global threshold. And I used the 85% to classify sequences to phylum from the work of Ransom and collaborators. So I analyzed the stream code Metabarcoding data with the optimized thresholds and with the global thresholds. And then I counted how many OTUs I will find for each phylum. So here I plotted the number of OTUs, and this is both in log scale for the optimized thresholds and for the global thresholds. So you can see that for most phyla, you find more OTUs if you optimize the threshold than if you use the 85% for all of them. There were a couple of phylum found only with optimized thresholds and one found only with global thresholds. And then I also look at the number of unidentified OTUs. And here you see the opposite pattern. There are more sequences that remain unidentified when you use the global threshold versus using the optimized threshold. So the take-home messages are that if we have more sequences, we can help reduce the errors and unknowns in taxonomic assignment. And if we use taxa-specific optimized threshold, we can increase the number of identified sequences. And of course, this will be also for each taxonomic level. I just show you phylum today for the sake of time. Finally, some other steps that we can take in general to improve taxonomic assignments. One is to form interdisciplinary teams that someone already mentioned today, and of course it should include a taxonomist, to generate training opportunities in bioinformatics and these type of tools that are coming up very frequently nowadays. To know your reference database, it's important that you really study the database you're using to know its strengths and its weakness. Using multiple genetic markers has been proven by many researchers that it will increase species detection. And finally, if journals that publish a species description could require a barcode associated with the species description, as some journals are doing, that also will help us to get good quality sequences associated with a good description to populate these reference databases. With that, I want to acknowledge the symposium organizers, and I thank you for the invitation. My advisor, Karen Osborne, and the invertebrate zoology community, then our funding, and the NCBI taxonomic team that has been really, really helpful, and all of you for your attention. Thank you very much.
Yes, thank you very much. Very nice, very interesting talk. There are room for questions here. Torsten?
You showed that you could increase the number of identified sequences using the threshold approach. So that, for me, means the threshold was lower than the global threshold. Am I correct with that?
Yes. For some of them.
Yeah.
For some was higher.
Yeah, but for example, what sticked out there for me was the flatworms. And I know with flatworms, I mean, it's only anecdotal, but I often have problems to find something, even just a phylum. And there I wonder, how certain can you be that what you have now as additional identified sequences, that they are truly flatworms, for example, or whatever the additional ones are?
If you're using these database, yeah, you can never be certain, right? So I feel that for any kind of work with large data sets, you need some dose of healthy skepticism. That's what I said. You do need to know the reference database you're using, and hopefully that's why these thresholds and suggestions need to be specific to the database. So it's as best as we can do. I'm adding I didn't have time to analyze it for this talk, an analysis of novelty to see if you have a novel phylum that is not represented in the database, what is the likelihood that you are going to make a mistake? But I think that for some of these, I don't know, the simulations that I'm doing, I think you can be fairly certain that you're not going to make a mistake. And the mistakes, I can detect mistakes in the sequences by looking at the matches. So if I see something wrong, I can find the mistakes, because you find a match that is like, this is really unlikely. And then if you dig into it, you can find an error like the one I found. It was because it was a weird point in my data, and then, well, I realized the taxonomy was wrong in GeneBank.
Yeah, it's not necessarily the taxonomy is wrong. The case I had, we know it was a flatworm. We had CO1 and 18S, and we blasted against NCBI with BLAST-N, both, and we got back with CO1 and annelid, because flatworms, it seems to me, still really poor with CO1 representative in NCBI. Or, yeah. And when we use the 18S, we got the one we ID'd morphologically back 100%. And I think for phylum assignment, family assignment, 18S is probably the much better marker to use, at least for many invertebrate groups, because due to deep level phylogeny studies, you have a much better representation of 18S and CO1 in there. And then we're speaking about very deep times, CO1 can get very different. I had the same with Stiocarpendella. USA one time came into my office and said, oh, it's an arthropod, I have a contamination. No, you haven't. Because there was, at that time, not enough annelid. So the CO1 blasted back against an arthropod. When we did the tree, it was perfectly fine annelid. In the tree, it mapped perfectly. And so I think that the threshold, just getting more sequences assigned might not necessarily be good, because then what you show it, you might get into the false negative rate much higher.
Yes. yes, that's why the number of sequences is also important. And we are doing this for different markers too. So that will come up for sure, that the other markers are better for phylum, yeah, or for the higher levels.
Any more questions? I have one comment. I was very glad that you noticed that the various reference databases gives different results, because people are not aware of that. We work with stakeholders, and they sometimes you find very strange results in the fish communities, and we try to explain this, why this is the case. But it's really an important issue. But I don't think people are aware of this, really, that it really depends. And so thank you very much for this.
Yeah.
So the last talk here for today is by Professor Bent Edvardsen from University of Oslo, Department of Biosciences. And in this case, I think it's going to be more applied metabarcoding study. Okay, thank you.
Yeah, thank you. And thank you for the organizers for the invitation to talk here today. So I'm going to talk about how we can use metabarcoding and systematics to study Protists diversity in the ocean and to better understand ecosystem functioning. And in order to, before I start, I would like to acknowledge my former or present colleagues that have contributed to much of the work that I will present. Eliane Egge, Sandra Gran-Stadnichenko, Lukas Upraha, and Wenche Eikrem. And the massive loss of biodiversity due to human activities is accelerating. And in order to conserve biodiversity, we need to know the organisms, who they are, their distribution in time and space, and their ecological role. And to predict the consequences of human activities and mitigate losses, we need to understand driving forces and processes that shape biodiversity and the community structure. And by integrating several methods, we can obtain a more detailed understanding of the community composition and distribution and ecological function. And in my talk, I will show how we can combine different methods and to better understand this ecological function, and also point at some possibilities and challenges. So what are protists? Protists, they are all eukaryotes that are not animals, fungi, or plants. And they are usually small, single-celled organisms and much less studied than their macroscopic eukaryotes. Although they dominate the diversity of eukaryotes. And they have a wide range of sizes and diverse forms and lifestyles, and play key functional roles in all ecosystems. And their nutrition can be photosynthetic, like the algae. They can be heterotrophs, like the protozoa, or they can be mixotrophs, which is everything in between. Many are symbionts, mutualistic symbionts, and many are also parasitic. And phytoplankton, they have photosynthesis, so they are primary producers and the basis for most food webs in the ocean. And they have been amply studied, both the diversity and production, in order to assess the potential fish production and also fisheries, harvesting of fish. And phytoplankton varies a lot through the season, both by the species composition and also abundance. And this has been studied for a century. And at University of Oslo, H.H. Gran and Trygve Brårud were world-leading pioneers within phytoplankton ecology. And they and their students, they studied seasonal dynamics and distribution and related this to environmental factors to understand the drivers of the ecosystem structure. And also Grete Hasle and Øystein Påske became world famous scientists in this field. And this picture shows the plankton room at the section for marine botany at the University of Oslo. And here you can see Professor Trygve Brodin with his students and colleagues counting phytoplankton cells under a microscope. And this is actually the same method that is still used in monitoring in Norway today. It's the standard method in Norway and elsewhere. And these data that they produced have been compiled, and they date 100 years back. It's phytoplankton and physical and chemical data from the inner Oslofjorden, and they compiled together with more frequent monitoring data from the last 40 years, and in a database by Elisabeth Lundsør et al. And these time series are extremely valuable in order to be able to reveal changes in biodiversity over time, and also in order to understand the environmental factors that are other drivers. So in another paper by Lundsør et al., we have used this database to study the changes in the phytoplankton community in the Oslofjorden. And this picture is showing the phytoplankton cell concentration over the last 90 years, how it has changed. But since a decade, the Metabarcoding has become the standard methods for biodiversity studies of protists and other microbes. And this is because it provides high taxonomic resolution and may detect also the tiny and fragile and rare species. And it has also the potential to be more automated and time-saving, and can also allow more frequent sampling. And the 18S ribosomal DNA RNA gene is the most commonly used marker for protist. And here we find the best reference sequences and the most reference sequences, and also well-curated databases, the PR2 and the Silva. And the V4 region of the 18S is variable enough and long enough to get a resolution enough to separate at the species level in many cases, but not in all. And the high throughput sequencing technology that are mostly used today is the Illumina MySeq pair end technology that gives about 500 base pairs reads, which fits to the V4 region. And there are also emerging long-read technologies that have started to come in use, that can cover the entire operon of the ribosomal RNA. I come back to that. And when we do metabarcoding of protists in the sea, we start with collecting waters from different depths with a water sampler from a research vessel. And we collect the plankton by filtration on the filter, and we extract all the organisms on the filter, the DNA or the RNA, and we amplify the marker gene by PCR, do some preparations, and do the high throughput sequencing. And we typically obtain between 50 and 100,000 reads or sequences per sample. And then we have to do lots of bioinformatics and biostatistics to analyze this big data. And to illustrate what sort of data we can get, I will show some examples based on a sampling campaign that we have done in the Outer Oslofjorden monthly during 2 years. And in addition to collecting water samples for molecular data or for metabarcoding, we also did measurements for physical parameters, for collecting water for chemistry, water chemistry, and also collected samples for microscopy and algal culturing. And the question we ask, some of them are the same as 100 years ago. So what is there? And how is the seasonal and vertical distribution of different taxa? And how vary community composition by season, and how vary alpha and beta diversity by season, and what environmental factors are main driving forces for the observed community structure, and how vary ecological functions in the protist community through the year. And the Metabarcoding have revealed massive unknown diversity, and especially of tiny cells, fragile symbionts living inside other animals, for example, or other protists. And we have had a project focusing on the haptophytes. It's a phytoplankton group, and they are very important primary producers in the ocean. Some form massive blooms, and some of these blooms are toxic and can kill fish. So they are also economically important. And they are small, so they are difficult to identify in the light microscope and are often overlooked in monitoring. So what we did, we really revealed a large diversity of haptophytes in the Oslo Fjorden. And this is the phylogenetic tree that shows how the haptophytes are related to each other. And in yellow, you see sequences that are from a cultured, identified known species, what we call a reference sequence. And in red, we have the OTUs, the gene types that we have obtained with the Metabarcoding. And then the black, they are environmental sequences from other places. And what we can see is that some OTUs, they fall close to a reference sequence, and then we can identify them to a species. But most of them, they fall in clades where there are no reference sequences, as you see here. And these OTUs may represent a novel genus, these may represent a novel order, and these may represent several new classes, novel classes. And we don't know what they look like because there are no reference sequences in the same clade. But recently, a new class was described. The HAP5 got a name and got one new species. So this is actually a new class. And when we opened another box in the same tree, a box containing, these are the sort of closed boxes, the boxes containing species of the genus Cryptic, we also found a large diversity and large unknown diversity. And Cryptic, they are tiny flagellates that look very similar in the light microscope, and we need to identify them in electron microscope to species, such as this Cryptic ledbeatery, which caused massive fish kills just 3 years ago in Norway. And what we can see here is that there are few reference sequences within this group, Cryptic, which is also a family. And the reference sequences are here in blue, whereas there is a large diversity of OTUs that have not yet been either sequenced or not even described yet. Oops. And there is also a large diversity has been revealed, but also massive data on species composition and distribution by season and space has been obtained by Metabarcoding. And this is a figure also of the haptophytes in Oslo Fjorden, how the seasonal or species composition varies through 2 years. And we can also see that the species composition, or the different groups that have different colors here, have different preferences through the year. And for example, this taxa here in pink, representing the Coccolithophorium Liania huxleyi, typically dominate during autumn, whereas others have a preference of being dominating in the spring. And also richness typically varies through the seasons, and the lowest richness was found here in April during the spring bloom, which is also commonly found for other groups. And we found that the highest richness was found in August. But there are also many challenges with metabarcoding, and we have heard some already. Molecular lab work and sequencing introduce both errors and biases, and such as primer bias, chimeras, errors that are not, diversity that are fails. And by better protocols in the lab, but also bioinformatics, some of these problems have been reduced. And also different bioinformatic pipelines give different results, as we heard, just heard. And there is still a big need, a great need to standardize the bioinformatic pipelines further. And also another problem or challenge is that the marker gene copy number differ between taxa, so which means that it's difficult to quantify these data based on metabarcoding, because some can be better represented than others. And also with the Illumina sequencing that is mostly used today, we get short reads. And some taxa are identical in the 18S ribosomal RNA. So we can't separate them. So then the long read sequencing has started to emerge. But they have other challenges, for example, the lack of good reference databases for long reads, and also the pipelines that are not that developed yet. And other challenges is the quantification. At best, we can only get relative abundance and not absolute abundance. So how do these metabarcoding data then compare to traditional microscopical cell counts? Do they show the same or not? And also another challenge is the lack of sufficient reference sequences for taxonomic classification. And I will then further point and discuss and show some examples how we can deal with the two last points. So in the study of Protists in the outer Oslo fjord, where we studied all the different Protist groups, we compared the quantification ability with the metabarcoding and microscopy cell counts. And we compared the relative abundance, where we made a ratio between reads measured by metabarcoding and biovolume obtained by microscopy. And we first looked at the class level. How are the different classes represented in with the two methods? And when the ratio is close to 1, the two methods represent the group equally, rather equally. And the two major phytoplankton groups in our waters, the diatoms and dinoflagellates, they were rather well represented both in microscopy and by metabarcoding. All the diatoms a bit underrepresented in metabarcoding, and dinoflagellates a bit overrepresented. But some other groups, they were largely underrepresented by microscopy, overrepresented in metabarcoding. And these groups, chrysophytes and dictyocophytes, they are mostly small, fragile, difficult to identify in the light microscope, so they have not been identified even to class level in the microscope. And this group here, the euglionophytes, they were not detected by the metabarcoding due to primer errors. So these, but they are large and easy to identify in the light microscope. When we compare the two methods, also the metabarcoding and the light microscopy over the season, we could see that the two major groups, the diatoms in red and the dinoflagellates in blue, they showed rather similar pattern. We get the peaks of diatoms and dinoflagellates at the same time. But the other groups are not corresponding, as we could see here to the left. But when we look into each class, for example, the diatoms, we could see that the species are different that we detect with the two methods, because we more easily detect the small but well represented species that have been sequenced, whereas here by microscopy we usually can identify mainly the big cells, but not always with the reference sequence. So our conclusions from this is that we need both methods in order to be able to reveal and get detailed understanding of the taxonomic composition and the richness and the abundance. And that we need both also in order to be able to link historical data, which are very valuable to use with more modern monitoring. And the next challenge is the lack of reference sequences. And in most protist groups, only a fraction of the described species has been sequenced. And with haptophytes, about 300 species have been described morphologically, have a name, but only 100 of these have been sequenced in the marker range, like the ribosomal DNA or ribosomal RNA gene. And by environmental sequencing, it has been revealed 900 different genotypes in the 18S that may represent potentially 900 haptophyte species. So in order to be able to identify these 800 remaining species, we need more cultures, more species descriptions. And in order to understand better the phylogeny and also revise the taxonomy in Haptophyta to obtain more monophyletic taxa, we have been isolating haptophytes, kept them in cultures, sequenced them, characterized them in electron microscope, and described species for about 25 years. And with an arrow, we have marked some of the species that we have described. And this has been done in collaboration with international scientists. And here are also two of the species that we have described on the haptophyte. So there is a large diversity still that we need to discover and describe for taxonomists, and we steadily find new species that need to be described. So there's a lot of work to be done for taxonomists for a long time within protists. And in metabarcoding studies from the Arctic, it has been shown that there is a large unknown diversity and also a great lack of reference sequences. So in the project Taxonomic and also in Nansen Legacy, the aim have been to improve the reference sequence databases. So we have isolated about 250 microalgal strains that have been cultured, characterized in microscope, sequenced, and done systematics on. And these has been isolated either by cell picking under the microscope or dilution series if they are abundant and tiny. And these strains are now maintained in the Norwegian culture collection of algae. And this culture collection is the largest collection of living algal strains in the Nordic countries, when we have more than 2,200 strains available. And it's owned and maintained by NIVA and University of Oslo together. And the cultures are used for research, teaching, innovation, and commercial use. And they are they are available from our webpage for everyone. But with these cultures, we can do a lot. We can study the morphology, and we can sequence several genes and long reads to get enough information so that we can do proper systematics and describe new species. And in the study by Supraha et al., just published a couple of weeks ago, what we could see was that many of these strains in red here that we have isolated from the Arctic, they are actually very similar to another diatom that are present in temperate waters, but genetically separate. So here we talk about cryptic species. They are morphologically very similar. We thought they were the same species, but here when we sequence them, we see that they are either a separate species or a separate variety of these diatoms. And the Metabarcoding is a recently published database of 18S rRNA metabarcode studies. And it contains 41 metabarcoding datasets corresponding to more than 4,000 samples. And all the data has been reproduced through or reprocessed through the same bioinformatic pipeline so that they can be compared. And this database is produced by Daniel Volo and his co-workers. And this, by using this database, we can get information about the biogeographical distribution of a genotype, and we can also get information about under which ecological conditions, environmental conditions, have this been found before. And so with these strains that Lucas Supraha has then isolated and characterized from the Arctic, he also analyzed their biogeographical distribution by using the Metabarcoding database. And what we could see then was that some of these diatom strains had a cosmopolitic distribution or a more temperate cold distribution, whereas especially those that, others were more restricted to the Arctic. So one question is then whether these strains or genotypes that are restricted to the Arctic, whether they are also more sensitive to climate change and warming and reduced salinity. So for that, we need to do experiments. And we need to do growth experiments to test the growth preferences and tolerances under different environmental conditions. So in our lab, we have set up a high-throughput growth response setup and in a factorial design so that we can test several parameters in combination. And here we have a plate with 96 wells where we can grow our strains in these small wells over time under very variable conditions. So this can tell us about the physiological response. And we have done this for some of these strains from the Arctic, but there is a need for much more of these type of experiments to get more trait information from these strains and species. Another trait which is crucial to know, because the protists, they can have different trophic modes, is to understand how the trophic mode varies through the season in a community. And this can also it's implemented, the trophy is implemented in the Metabarcoding database presently, and we have assigned each OTUs, each genotype, to a trophy. We have assigned it to an autotroph, a phototrophic organism, a purely heterotroph, or a dinoflagellate. And the dinoflagellate, they can be either heterotroph, phototroph, mixotroph, parasitic, mutualistic symbionts. So to distinguish, to separate them into different groups is more complicated. So what we could see is also the trophy is not surprisingly changing through the season. And what was maybe interesting was that the purely heterotrophic groups, they are represented quite equally through the year. Whereas during the winter time, when the phototrophs are less abundant, they are replaced by the dinoflagellates. different types of dinoflagellates, probably. So trophic mode is one trait that we can be interested in to understand more the ecological function of the community. So to be able to link then taxonomy, I mean, go from a DNA sequence to a taxonomic name, and then understand that the community's ecological functionality, then we need to know the organism. When we need to know the traits, we need to know trophic mode, motility, habitat, life cycle, physiology, interactions with other organisms, growth rate, size, form, etc. So this information we need to obtain by different methods. And this information can be found to some extent, some of it, in the literature, but a lot of it is missing for protists. So we need to produce this data by different methods, such as experiments, systematics, genomics, and combine them to and include these data in our databases. So there are already many labs that are producing trait tables for their localities, but these will eventually also be implemented in more general databases. So in summary, no method can fully describe the biodiversity alone. So by combining several different methods, a more detailed understanding of the community composition and distribution and ecological function can be obtained. And I would like to also thank my colleagues and collaborators and funding and you for your attention. There.
Thank you very much for a nice talk showing how it can be used, metabarcoding.
Mm-hmm.
It's time for one question. It's a bit late, but it's one question. Okay. I had a comment, but I think you answered that. It's. Do you want to make a question? No, go ahead. Okay. You showed, so you can find many more taxa using metabarcoding than with the traditional methods. Quite amazing number of taxa, yes. Okay, one quick one then.
Sorry. Thank you. The tree where you showed, where you mentioned that some species turned out to be probably cryptic species. I don't remember exactly which taxon it was. I saw that there were then, so you indicated the number of potential cryptic species in red. Then the clades that fell in between, would you have called these phenotypically close to that complex of cryptic species, or
well,
did it surprise you that they clustered there, or?
It surprised me that we actually saw that they were genetically different, although we thought they were. It was diatoms, for example, Chaetostros species, especially Chaetostros species. It is a big genus, maybe several hundred species in the genus that have been studied thoroughly, but that they actually have, Chaetostros debilis was one of them that turned out to be separated clearly in two clades. And one clade were restricted to the Arctic. And that was also what we could see for other species. Not all of the red one were cryptic, but we could see several species that, or if it was species or maybe varieties. Yeah.
Okay, thank you.